actually. Everybody, good morning.
Morning, sir. Just a few minutes.
Morning,
I'm trying to go live on YouTube.
Okay, sir.
You're live on. Ramesh? Ramesh?
Yes, sir.
The link I've shared.
Yeah, we got it. We got the link. It's there.
YouTube link is there. Kindly share to all.
Yeah, please, sure.
I think the speaker is already in. Shall we start, sir?
Sure, let us go ahead.
Dr. Sanjay Saab?
Yes, sir. We can. Yes, sir. We can start.
Link I've shared. Dr. Sanjay,
Yeah,
sir.
we got the link.
Sanjay Chibba, are you there? Link is there. Kindly share to all.
Yeah, please, sure.
I think the speaker is already in. Shall we start, sir?
Yes, sir.
Yes, I think you should. Let us go ahead.
Yes, we can.
I think it is echoing because even the YouTube, I think someone has on it through the Zoom, because it takes around a 30-second gap. So kindly mute the YouTube. That's more important. John, welcome.
Thank you.
Thank you, sir. We'll start the program. At the outset, good morning, everyone. At the outset, I welcome all to the second day of the second International Conference on Bacteriophage Research, organized by Society of Bacteriophage Research and Therapy. It gives me immense pleasure to invite and welcome the President of the SBRT, Professor Sanjay Chibba. Is an Emeritus Professor, Department of Microbiology at Punjab University. Welcome you, sir. It's a pleasure to invite the Secretary of SBRT, Professor Dr. Gopal Nath, sir, is a professor in the Department of Microbiology, Banaras Hindu University. I welcome all the core committee members, editors of our journal, Dr. Ramesh, core committee members, Dr. Urmi, Dr. Sangeeta, Dr. Taruna, Dr. Ajay, and I welcome all the delegates, moderators, chairperson, and the keynote speakers. I welcome Dr. John for this conference. I request Dr. Gopalnath and Dr. Ramesh to chair the session. A brief about Dr. John Dennehy. Dr. John is a professor of biology at Queens College of the City, University of New York. Dr. Dennehy trained in phage biology as an NSF postdoctoral fellow in the Turner Lab at Yale University and the Wang Lab at the State University of New York at Albany prior joining the faculty of Queens College in 2007. Dr. John has published over 50 articles and contributed in book chapters, conference papers on phages and the virus evolutionary biology and ecology. He is also a senior editor of Microbiology Resource Announcement and an associate editor of Virus Evolution and curator for the American Society of Microbiology COVID-19 Research Registry. It's a great honor to have you here, John. So kindly, please, you can go ahead with your presentation.
Thank you very much for welcome. It's about 12:30 a.m. in New York City, and I wish you all a good morning. It's technically morning here now. Let me bring up my slides. Are you able to see my title slide?
John, you need to share the slide. The slides are not visible.
Just a minute. How about now? Is it working?
Yes, John. Yes, John.
Excellent.
Yes,
So I would like to talk a little bit about the control of bacteriophage lysis timing. I consider myself an evolutionary biologist, a ecologist, but I do have a fascination with one of these. I'm sorry, I just pressed the wrong button. I do have a fascination with a particular molecule called holin. Holin is nature's membrane perforation tool. In bacteriophages, generally they have two molecules to disrupt the membranes and the cell wall. And these are the holin and the endolysin, of course. And also in this diagram, I want to share a couple of other molecules, OZ and OZ1, which are called spannins. The way the system works is basically holin and antiholin are expressed. They accumulate in the membrane, in the inner membrane of the cell. And at a genetically encoded time, which is very precise, they form a hole. And this allows another molecule, endolysin, to access the cell wall and dissolve that. And then subsequently, the proteins RZ and RZ1, the spannins, will disrupt the outer membrane. And then you will have lysis and the bacteriophage progeny are released. I'm sure you all know all this. But one thing that I think is very important is to understand how this system is regulated and controlled, because I think in the long-term future, in order for phage therapy to really be employed broadly, we are going to have to engineer phages. And to understand the phages we're working with, prerequisite for properly engineering them. Just to show you the lysis process, here is a cell undergoing lysis. And one of the things you notice here is the cells start to deform from a rod shape to more of a sphere. And this is actually where the cell will lyse. But that deformation is first off, the holon is being released, forming a hole, the endolysin dissolves the cell wall, the cell then loses its rigidity, and then the pressure of the cell itself will cause the cell to burst open. Here I have another video which shows GFP tagged holon and its activity in the cell membrane. Here's E. coli with GFP tagged holon being expressed. And you see over time, at least initially, the holon is fairly evenly distributed about the cell. But there's a moment when, I'll show right here, where all of the holon is starting to coalesce. And usually this happens at the poles of the cell where the holon will accumulate. We call this nucleation, sort of like crystal formation, where these holes will form generally at the pole. So this is a very interesting process that kind of undergoes a phase transition from one state to another. And now I want to look at the kind of the way holon and antiholon are expressed. This is from the classic Bacteriophage lamba. Bacteriophage lamba, when it is induced into the lytic cycle, essentially the gene making up what we call the lysis cassette consists of the holon molecule, S gene, and R gene, which is endolysin, and then hispanins, RG and RG1, are all under the control of the same promoter called PR prime. PR prime is actually a constitutive promoter, but there is an anti-terminator, a terminator that interrupts the transcription of what we call the late gene, the lysis cassette, and all the morphogenic genes that make up the phage structure. Soon after induction there's another protein produced called protein Q, which is an anti-terminator and frees up transcription of the S gene and the other lysis protein. However, in some phages, lamba, many of the lamboid phages, they have actually what we call a dual start motif. And there's actually two start codons for the same gene. And we get two different proteins produced with diametrically opposite effects. The first protein is the 105 amino acid, which we call holon, and the second one is a 107 amino acid protein, which we call antiholon. The only difference between the two proteins is that the 107 amino acid antiholon had an extra couple of amino acid residues by virtue of starting sooner on the transcript than the 105 amino acid holon. Access to these two different start codons is controlled by a hairpin that forms in the Shine-Dalgano region of the mRNA transcript. When the hairpin forms, these residues will bind together through complementary binding, and the RNA polymerase will be unable to bind, the ribosome will be unable to bind to the first start codon, and it will then bind to the second start codon and make this 105 amino acid holon. Every now and then, though, the hairpin will relax and access to the first start codon is permitted. And then the 107 amino acid antiholon will be translated. And this happens about one time for every 2 times that the holon is produced. So you end up with one antiholon for every 2 holon that are produced. Now, what's very interesting about the holon and antiholon is that they both are membrane proteins, and they insert themselves into the inner membrane of E. coli. The holon will take up a 3-transmembrane domain conformation with the N-terminus in the periplasmic space and the C-terminus in the cytoplasm. By contrast, the antiholon is unable to take this 3-transmembrane domain shape. The reason is one of those 2 extra residues that are possessive is a lysine, which has a strong positive charge, and this prevents the M-terminus of the protein from crossing over the membrane, because the membrane is negatively charged. So it gets stuck in this 2-transmembrane domain shape. Nevertheless, it can dimerize with holon and inactivate it. So for every 1 antiholon produced, it will bind to 1 holon. And for some reason, we don't quite understand antiholon and antiholon tend not to dimerize together. Instead, antiholon will almost always bind to holon. And as a consequence, the 2 molecules, holon and antiholon, are functionally inactive initially. The other holon that are produced will dimerize, insert themselves into the membrane, and continue to accumulate until a genetically encoded time in which then they will trigger the lysis process. And here's kind of a cartoon showing how this works. Holon and holon will dimerize, and these will be functional. On the other hand, the antiholon and holon, when they dimerize, they will be non-functional. So initially, this holon and antiholon accumulation will proceed and continue dimerizing in the membrane, and it'll be evenly dispersed in the, as I showed you in the short video. At this point, the proton motive force is strong. It had a strong negative charge. The holon dimers will accumulate into the membrane, and then at a certain point, they will undergo some sort of phase shift. When this critical concentration is reached, they will start to form what we call rafts, and then these rafts will orientate themselves such that the hydrophobic areas will face out and the hydrophilic areas will face in, because remember, they're embedded in the membrane, which is hydrophobic. Once this pore has formed with the hydrophilic faces facing in, they will form literally a tunnel through the membrane, which then allows hydrogen protons to flow back down their concentration gradient, and rapidly the proton motive force dissolve. So you have a depolarization of the membrane, which then allows the antiholon, which is stuck in this 2-transmembrane domain state when there is a proton motive force. When that proton motive force is removed, this first transmembrane domain can collapse across the membrane and create this 3-transmembrane domain status, just like the holon does. So functionally, once you remove the PMF, the antiholon can contribute to hole formation, and the hole will get bigger and bigger, and eventually you'll make a micron-sized hole, which the molecule endolysin can leak through and dissolve their substrate, the cell wall. And then thereby the cell will lyse. So this really brings up some interesting questions to me from kind of a, thinking about the evolution of the system. Why would you make this antiholon when essentially it had no initial function in forming wholes? Proteins, we know, are the most energetically expensive class of macromolecules. So why spend all this energy to make a molecule that's going to inactivate 2/3 of the S gene, which is the gene that makes antiholon and holon? Presumably, this must have some sort of big benefit, which is not quite clear. And another fact that we must acknowledge is that the phages in which we've taken out antiholon are perfectly functional. In fact, they lyse a little faster than the phages with antiholon because they can accumulate holon up to the critical threshold much quicker. So in other words, this is some sort of kind of a Rube Goldbergian lysis. And maybe if you're not familiar with this, he's a cartoon artist back in the Great Depression, and he would invent these fantastic machines. Here's an example of a self-operating napkin. It has this really complex mechanism. The analogy here is, why do phages or some phages have this really complex mechanism when a simpler one will do? My predecessors, my former postdoctoral advisor in Nang Wang, had written at one point that antiholon thus appears to be a fine-tuning system for the scheduling of lysis. And his postdoctoral advisor, Ra Young, at the Texas A&M University, said that antiholon exists because it confers on the holon lysis system an all or nothing character. Unfortunately, I'll have to disagree with these two fellows. My collaborator and I showed that if you want to fine-tune the scheduling of lysis, all you need to do is to adjust what we call the translational burst size, not to be confused with the burst size with number of phage progeny being released, but rather the number of protein molecules made per RNA transcript. So you can adjust that number, or you can adjust the threshold concentration by which holon need to accumulate to trigger lysis. And that's adequate to fine-tune the lysis system. So essentially, you can do the fine-tuning without any holon. And also, we also showed that abolishing antiholon does not impair lysis ability at all, or diminish this all or nothing character. We find that the antiholon-less phage lambda is able to trigger lysis extremely efficiently and as quickly as does the wild type. So this really poses a kind of a big mystery. What's the reason why we have antiholon? And after thinking about this system for quite some time, I think I may have come up with a few reasons why. And to do so, I want to kind of just go over the assembly dynamics of what's happening. Once you have induction of the lytic program, about 12 minutes later, you have expression of Q, and that's a terminator protein that will enable the lysis cassette and the phage late genes making up the structural proteins all to be expressed. So these structural proteins start being expressed about 12 minutes into the onset of lysis. And at the same time, holon is being produced. And phages are being assembled in the cytoplasm. But interestingly, after about 25 minutes, there is sufficient holon available in the cell that we can artificially lyse the cell using potassium cyanide. So this implies that the amount of holin in the cell at 25 minutes is sufficient to lyse the cell, but even before any phage progeny have been produced, because the first phage progeny do not appear until about 30 minutes. So if the cell were to accidentally lyse too early, it would in effect be suicidal. The assembled phage have not fully assembled that they can be released at this time and infect other cells. So essentially the cell would die and the phage ability to make progeny would die along with it. So my first hypothesis is that antiholin will buffer against stochastic variation in gene expression. We all know that gene expression is somewhat noisy. It can be a function of the number of ribosomes, the number of RNA polymerases, the concentration of DNTPs, so on and so forth. All of these, if they're, should happen to be randomly a very high concentration, you might be very efficient in manufacturing holin and actually do it too quickly that you would lyse the cell before any phage progeny have been produced. So by making this antiholin, you're buffering against premature lysis without actually reducing the expression of all the other polycistronic structural genes. And we have a name for this kind of system, actually, one where you have transcription of RNA, which can then make an activator and an inhibitor at the same time. This is called an incoherent feed-forward circuit. Most often when we think of control systems, we think of feedback. We think that's the best way to regulate such systems. But my colleagues and I have shown that it's actually not the best way to regulate systems that are controlled by a very stable protein. And it ends up, if you have a very long-lived stable protein, it ends up being a lot more noisier with a feedback system than when you have a feed-forward system. Anyway, I have another hypothesis that might also explain the presence of antiholin, and that is it protects against premature cell death. So cells, as they're growing, the phage take over the cell, hijack the replication machinery to produce more baby phage. But if at some point the cell should die before that critical threshold of holin is produced, then the phages are trapped within that dead cell. What antiholin then does is provide a critical reserve that can be converted into holin when the PMF is abolished. So if the cell should die prematurely before there's sufficient holin present, that antiholin can then be converted into holin in order to lyse open the cell, even though the cell is dead, thus freeing up those trapped phages. Such conditions may not be common when we grow E. coli in the lab because of, we're growing them in a high nutrient environment with few stresses. But in the wild, perhaps it may be more likely the cell will inadvertently die in the middle of phage reproduction. So these two possible roles for antiholin are not actually mutually exclusive, and they may, in fact, complement each other, preventing early lysis by the rapid accumulation of holin, or by preventing inducing lysis if the cell should die prematurely. So thinking about this system, my colleagues and I used a combination of modeling and stimulation and experimentation to explore this system where we use the incoherent feed forward as a control mechanism to buffer noise and lysis timing. And our goal is to produce a comprehensive model of the relative influences on molecular noise and the impacts on phage life history, fitness, and burst size. So we model phage lysis as a first passage time process. This is something we borrowed from control systems and kind of the system biology of many other different processes. We apply it to phage biology. So briefly, as I mentioned before, you have induction of the PR prime promoter. This is going to, once this system is turned on, then you will have expression of the late transcript. We would say that this operon or cistron is turned on or turned off at a certain rate, K. The late transcript produced at a rate, Km. Translation of holin occurs at a rate, Kp. And the transcript is degraded at a rate gamma. The free holin is produced stochastically. So in some cells, it will be produced rather quickly. And if you trace these paths as holin is produced over time, you would have lysis early in the cell life. By contrast, some other cells might accumulate holin more slowly. Perhaps they don't have as many ribosomes or whatnot, and they would accumulate and reach that critical threshold at a later time point. And if you were to plot a histogram of these arrival times when the holin accumulation has reached this critical threshold, you will get this kind of bell curve kind of shaped curve. And this is reflecting how the threshold is reached at different times in different cells. So we can actually look at single cells and see what their lysis times are to estimate not only the mean of this histogram, but also the spread. And the spread is actually what we would call the noise. So we developed a mathematical model, which we model the mean first passage time, where we can have parameters for the threshold, the translational burst size, and to predict not only the mean lysis time, but also the coefficient of variation. This is the standard deviation over the mean, and it's a measure of noise in lysis timing. And through these models, we can generate predictions as to what would happen when you manipulate, say, transcription or translation, or you increase or decrease the threshold. And we made up several different predictions, and we created a couple of different models. This phage expression is kind of not exactly constitutive, even though the PR prime promoter is constitutive. We have that regulation by a terminator and an anti-terminator. So we do have a promoter switching model as well, which generates some of the same, but also different predictions. I'm not going to dwell too much on the prediction because most of them are wrong. We did model the how the ratio of Holon and anti-Holon will affect the noise and lysis timing. And what we do see is that when the Holon and anti-Holon expression is correlated, it minimizes noise. So that's something I'll come back to. But essentially, what I'm trying to show here is that we generated a number of different mathematical models, we generated a number of different predictions, and then we went about the process of testing the prediction. And then later on, with the experimental result, we went back and we changed our models. So a process of refinement based on the interaction of modeling and experiment. As the first step to testing these models, we generated mutated Holon molecules or Holon genes. And we systematically modified every residue in the 107 amino acid or 105, 107 amino acid Holon, anti-Holon, changing each residue in turn so we could see what the effects of these mutations on the lysis timing and noise and lysis timing. And to look at the single cell lysis time mean of distribution, I set up at a postdoc this experimental system. Essentially, I used an inverted microscope, and this is used to view cells that are enclosed in a perfusion chamber. Essentially, this chamber is a piece of plastic with a hole in the middle through which media will flow. And then I can cover up that hole with cover slip from microscope cover slip. And the cells can be fixed to the cover slip so that they'll stay stationary while I view them with a microscope camera. This whole perfusion chamber is placed on a heating plate, and phage lambda has a mutant called C1857, which is induced from the lysogenic state to the lytic state through a temperature spike. So when you raise the temperature from 30 Celsius up to 42 Celsius, the lytic program is triggered, and then we bring it back down to about 37, and we record the video, and over time, maybe about 60 minutes, 65 minutes, the cell would accumulate enough Holon to begin lysing. And later on, we go back in the video and we observe the cell as it lyses and record the time in which it lysed, and do that for all the cells in the population. And through this, we come up with a mean and a distribution of lysis times. And one of the first things we noticed was when we lysed a whole bunch of these Holon mutants, the ones with Holon and anti-Holon tended to express far less noise in lysis timing, regardless of what the mean lysis time was, than did Holon without anti-Holon. So these cells in which we removed the anti-Holon lysed with much greater noise than did those with Holon and anti-Holon. In addition, we looked at the mean first passage time noise for a whole bunch of different Lambda mutants that varied over a very wide range of potential lysozymes. So the interesting thing is, by just changing one or two residues in the whole protein, you can speed up the lysis time or you can delay it considerably. And what we noticed was we see the kind of concave up-shaped curve. Going back to my predictions, when we increased the mean first passage time, the initial prediction was that the noise and lysis time should decrease, which is sort of what happens in the beginning. And the first couple of times we measured cells, we thought we were meeting that prediction, but then we started measuring longer and longer lysis times, and the opposite happened. So our original prediction that noise decreases with increasing threshold unfortunately need to be rejected, and we developed a new model which could very nicely predict the noise and lysis timing given a lysis time. One thing that's very interesting to us was that if you look here, the wild type has the lowest amount of noise in lysis timing of any of the genotypes. And, you know, we consider it to be the optimum noise. Additionally, we modified the promoter to look at how changing the promoter strength, making it weaker or making it stronger, would affect the lysis time and the noise and lysis time. It turned out to be kind of difficult to actually modify the promoter. And if you notice here, I only have 4 points on the line. And part of the reason is sometimes you modify the promoter, you get no expression. Sometimes you modify the promoter and it doesn't really substantially change the expression at all. Sometimes you modify the promoter and you severely abolish the expression. And regardless of what we did, no matter how many times we changed the promoter, we are never able to improve the performance of this PR prime promoter. So we never got greater expression than we did with the wild type. The mean lysis time tended to decline with increased expression, but after a time it sort of leveled out. So it seemed there's a maximum amount of expression from this promoter to produce Holin, and essentially after that point, you really can't increase it any further. And unlike our previous prediction, if you decrease translation, you should also decrease the amount of noise and lysis timing. That turned out not to be true. So if you decrease translation, you would actually tend to increase the noise. The other interesting thing about this noise and lysis timing is, this is a histogram. In the blue is the wild type, and I'm showing the amount of variation in lysis timing for the wild type. It's actually extremely small. So the amount of variation is about 5%. So the lysis timing for this wild type is about 65 minutes with a standard deviation of about 3 minutes. So about 3 minutes on either side of 60 minutes. About almost all of the cells were lysed. Even for this wild type, if you have an early lysing cell, based on the principle that the progeny assembly rate is linear, And for phage lambda, it's roughly produces about 7 phages per minute, starting about, this is not exactly correct. This is probably a little off. It starts about probably 30 minutes. The assembly of the first phage begins about 30 minutes and then accumulate linearly from there. But if you have an early lysing cell, lysing about 62 minutes or so, and you compare that to a late lysing cell, you can have half again as many progeny in the early lysing cell as you do in the late lysing cell. It's a tremendous difference for such a short difference in lysis timing. So that's kind of a clue of the importance of the precision in lysis time. If you consider that most of the phage in a population will be fairly genetically similar, if not identical, they'll all be lysing at the same time. And ones that are lysing early will have a slight advantage in getting to the next host cell, but they will suffer a fairly large cost in total output. By contrast, one that lyses late will have a much greater advantage in the number of progeny it produces, but unfortunately, it's going to be delayed somewhat in being able to diffuse to the next host cell because early lysing competitors will have slight time advantage. One of the next things that we did was we looked at how different phage genotypes, the different in lysis time, how that affected their fitness. And the way we did this was we would do serial transfers, and we call this kind of a semi or quasi-continuous culture. A continuous culture system is a chemostat where you have an inflow of media into a flask in which phages are infecting bacteria, and there's another outflow at the same rate, so that the phage and bacteria have a constant supply of nutrients, and there is removal of the waste product and phages and bacteria removed from the chemostat at a constant rate. The drawback with this kind of system is that they're really hard to operate, and you can't really set up too many of them at the same time. I mean, it's something we do in the lab, and maybe we have 4 or 5 chemostats set up at the same time. But if you want to do a properly replicated experiment using a lot of different genotypes, it's not really feasible. So what we did was we took the flask and we would culture phage and bacteria together. We start off with an initial number of about 10,000 phage and 10 million bacteria, and you would let them co-culture and grow for about 3 hours. So, initially the phage would be vastly outnumbered by the bacteria, but then they would reproduce through a couple of rounds of reproduction. And, at the end of 3 hours, they pretty much would have infected all the cells in that culture.
John, you have 2 more minutes.
Sorry, what was your question?
You have 3 more minutes, John.
Yes. So anyway, we would then take that flask and we would filter out the bacteria, any bacterial debris or whatever, and then dilute it by 1,000-fold. This is the phage lysate diluted by 1,000-fold, and we would add it to a fresh culture of bacteria. And we would continue this process through about 5 rounds of serial transfers. At every passage, there would be a 1,000-fold dilution in the number of progeny. So the phage must be growing at a rate of greater than 1,000-fold in order to avoid extinction. And after that 5th passage, we measured how many phages were in the culture. And here are our results. So every passage, we measured the number of progeny phage in that culture. And what we see is that the phages with a very long lysis time or a very short lysis time would be driven close to extinction across 5 passages. They didn't quite go extinct, but after 5 passages, we didn't really want to continue the experiment because you would start to see evolution. And these phages with a long lysis time would evolve a shorter lysis time quite quickly. So to avoid the confounding effects of evolution, we stopped the experiment after 5 transfers. So the very short and very long lysis time phages had a very severe fitness defect. By contrast, those phages with a lysis time of about 69 minutes or 65 minutes did quite well. I mean, we kind of stacked the deck in favor of these mid-range lysis times, but nonetheless, this experiment demonstrates that the lysis time had a big effect on fitness. And especially interesting here is that the wild type actually has one of the highest fitnesses of all phages, of all phage genotypes that we tested. And you can see here, it demonstrates under these conditions that there's an optimal lysis time in which the phages maximize their productivity across 5 passages. And this figure is on a log scale, and if you look at it on a linear scale, it's a much more dramatic peak. So to kind of summarize what we found is that the phage-lambda Holon lysis system, we believe, has evolved to minimize noise and also to maximize, to lyse at the proper time such that they can maximize the fitness of the phage. And some of the evidence that support this idea that noise minimization is very important to phage, we initially didn't expect it to be so important. We were just kind of interested in understanding, control mechanisms of gene expression. But when we find that the wild type has minimal noise and changing that noise altered its fitness considerably, and in addition, there are other supporting evidence such as the high transcription rate. The late transcripts are produced extremely rapidly, and each mRNA is only used once or twice before it degrades. The translational efficiency is also quite high. Not the efficiency, the promoter activity. I'm sorry. The promoter activity is almost as high as could possibly be. We weren't able to improve it through site-directed mutagenesis. However, the translational burst size, this number of proteins, the actual protein made from each transcript, the translation was extremely inefficient. You only get a handful of Holon molecules per transcript. The feed-forward control via antiholin also acts to minimize noise in lysosome. And holin and antiholin expression turn out to be highly correlated, which I showed previously that this high correlation between holin and antiholin expression minimizes noise. And no matter what we did, we systematically mutated every codon in the holin gene, and despite thousands of different variant genotypes, we were never able to find one that has less noise than the wild type. So interestingly, even though it's been a lab organism for over 70 years, Esther Lederberg discovered phage lambda back in 1949, 1950 or so. And, despite being carried in a laboratory all the time, well, perhaps because of it, wild type lambda had a minimum gene expression noise and an optimal lysis time. So it would appear, at least in laboratory conditions, that there's a very strong selection to maintain reduced noise and an optimal lysis time. So that's it for my talk, and I just want to acknowledge my collaborator, Sharon Cannelly, is a postdoc in my lab. He was assisted by an undergraduate named Anna Gao. My main collaborator, Abhi Singh from the University of Delaware, and Kem Gusinger from the University of North Carolina, In-Nang Wang from the University of Albany, Supravat Dave from the University of Delaware, Pavel Boaks from Comenius University. And I'm happy to take any questions if you should have any.
Thank you very much for a very impressive talk on control of bacteriophage lysis timing, emphasizing on the cascade of lysis timing in respect to holin, antiholin, and emphasizing on the stochastic variation in the gene expression of holin and the antiholins. Due to time constraint, and I will allow only 2 questions, I request the chairperson to moderate the discussion.
A very wonderful presentation, excellent information. And audience, do you have any questions? Since we are running out of time, so there is only limited questions. Any questions?
Yeah, I have a question. Hi, John. Excellent presentation. We learned a
Thank
lot.
you.
Very, very well. I just had 2 quick questions related. One, that when you did this experiment of serial dilution and you found that the long lysis phages switched to short lysis time during the 5 rounds of your experiment. So did you check back that what kind of mutation happened to put them back into the short lysis phase? And my second
Yeah.
question is that, given the role of antiholin you have explained in lambda so well, in therapeutic phages for whichever bacteria, if similar system operates, so then do you think that if you mutate the antiholin, so the lysis may not happen, and hence the phages, though they kill the cells, but they remain trapped inside the cell. So, with the fear of lysis of bacterial pathogen in the host is less, and release of endotoxin may not happen. So given the classical seminal work you have done on antiholin-holin system, can we leverage it in phage therapy? These are my 2 questions, please.
Yeah, I think that's a very good point. So we were able to generate lambda phage that produced so much antiholin that they effectively would not lyse the cells. And I think in terms of phage therapy, it could be very useful to engineer phages. And if you want to engineer a phage, when you start manipulating different components of the phage and the phage genome and the promoter, you'll have to at some point confront, how are you going to lyse this cell? By understanding, the regulatory mechanism of how cell lysis happens, you can fine tune the lysis and modify the phage, whatever phage that you have. Presumably one that has a very short lysis time, you can make it lyse longer or shorter, whatever the condition that would be optimal for your phage therapy.
Okay, thank you so much.
Okay. Any more questions for this for the talk? I think we can move next.
Thank you, John. Thank you for your time. In the late hours, we really appreciate, the society appreciates, your effort, in emphasizing the importance of controlling the bacteriophage lysis timing. Thank you, John, once again. Thank you.
Okay, thank you.
Thank you, Dr. John.
Thank you, Dr. John, for the very informative talk. Okay, we now move to our next speaker for the session, and we are extremely delighted to have with us Dr. Jeremy Barr. And it is my honor to introduce Dr. Barr to all of you. Dr. Jeremy Barr is the head Bacteriophage Biology Research Group and Senior Lecturer, School of Biological Sciences, Monash University, Australia. Dr. Barr completed his PhD in microbiology at the University of Queensland in 2011 and later completed postdoc at San Diego State University. He then moved to Monash in 2016 and established his independent lab called the Barr Lab on bacteriophage biology. In 2020, Dr. Barr joined Monash Center to Impact Antimicrobial Resistance. His lab's research interest includes studying the tripartite symbiosis between phage, bacterial host, and eukaryotic cells. And his work on developing the BAM model helps us to give very useful insights into how phages adhere to the mucus and help reduce colonization. With numerous research publications, close to 50 to his credit, he's a recipient of the Early Career Scientist Award 2017, SDSU Presidential Scholarship 2015, Teaching Excellence Award 2010, and GSK Student Excellence Award 2010. Actually, the list is never ending. So we welcome you all, you on this platform today, sir, and we now request Dr. Jeremy Barr to present his talk. The topic of his talk is bacteriophage coevolution with a mammalian gut mucosa. Welcome you, sir. Thank you. Over to you.
Thank you, Cindy. Thank you for the introduction. And thanks to everyone for the invitation and coming along to listen to me speak. Vanu, Ramesh, all my colleagues, really appreciate the invitation, and I'm very excited to present to you all. Give me a moment, I'll share my screen. All right, hopefully you can all see my screen and everyone can hear me okay?
Yes, Jeremy.
Awesome. It's really my pleasure to present here. I've always wanted to travel and visit India, so hopefully when travel resumes, I'll be able to come out and meet you all in person eventually. But until then, I hope you enjoy my presentation over Zoom. So my name is Jeremy Barr, and I'm Senior Lecturer at Monash University here in the School of Biological Sciences. And I'm going to present to you today some of our fundamental research. So we do a lot of work looking at phage therapy and translating some of these understandings. But today I'm going to talk to you about some really fundamental work that we're doing, trying to understand how phages co-evolve and really co-evolve in the presence of a mammalian gut mucosal system. And so I want to start my talk with this hypothesis or premise around tripartite symbiosis. And we know that bacteriophages and their bacterial hosts engage in a whole range of different symbioses. These can be strictly predator-prey, or they can be very mutualistic in the form of lysogeny. We also know these entities don't exist on their own, and particularly within the context of the human body, we know that there's a whole range of diverse interactions in the ways that particularly the bacterial component of the microbiota can interact with the cells, organs, and systems of the human body. This has been shown through the production of immune systems and maturation of the immune system, the production and secretion of short-chain fatty acids to influence the epithelial cells, and even the stimulation of this gut-brain axis where bacteria or the metabolites that they produce are able to influence our thoughts, moods, and potentially even the foods that we eat and crave. We also know that this isn't a one-way street and that the cells, organs, and systems of the body have a variety of mechanisms that they can use to communicate back and influence that microbiota. This can be in the form of metabolites, mucus production and secretion, or the secretion of antimicrobial agents. And if you look at this symbiosis, you'll see from the very traditional sense that the only way that phages can really influence the epithelial cells and systems here is via the bacterial host, by modulation of the bacterial host. And my lab is really trying to challenge this hypothesis, but looking at diverse ways in which phages can directly interact with the epithelial cells and system. So I'm going to present to you today some of our work looking at this in this tripartite system. So I want to touch on the experimental evolution of phages. I think we've got someone's microphone on. Maybe a little bit there. So looking at the experimental evolution.
Dear participants, can you mute yourself? Sorry, Dr. Jeremy.
Okay, no worries.
Participants, can you mute yourself?
Thanks, Ramesh. So looking at the experimental evolution of phages towards a mammalian host system. So there's been a lot of fundamental work looking at how phages and bacteria can co-evolve, and even how bacteria can co-evolve along with these mammalian systems. And what we really wanted to ask or probe in these experiments is, can these bacterial systems adapt and co-evolve directly within the presence of a mammalian epithelial cell system? And so some key points that I'll touch on. So we're looking at the phage adaptation to these tissue culture cells. We're looking at this in an experimental gut-on-a-chip system, which I'll describe to you in a moment. And we've done a lot of experimental evolution and sequencing work. And before I jump into this, I've really got to give a big shout out to Wei-Ho Chin, who is a PhD student in my lab, a senior PhD, and this is really all of his work that he's led. So I'm presenting his work, and a big shout out to Wei-Ho. Okay, so to start off, we really need to choose the appropriate epithelial system to work with. And our lab does a lot of work on static tissue culture cells, and these can be a really useful model system. They also have a lot of limitations. And one of the big limitations is that they can't support long-term bacterial co-culture. And this is due to the fact that the microbes will overgrow in the system, and that will crash that cell layer. We considered moving all the way through to animal models, and this obviously brings a lot of increased biological relevance, but it also increases the complexity of this system. And there's a lot of confounding factors, including the microbiome present within these animal models. And so we decided to use an intermediate system known as a gut-on-chip or lab-on-chip technology, where we're using engineering and microfluidic devices to simulate an organ-like system incorporating tissue cultures in this dynamic gut-on-a-chip system. And so I wanted to spend a couple slides covering this and summarizing this gut-on-a-chip. How do we make it? How do we operate and run it? And so really simply, we start off with a three-dimensional design file that we design ourselves. We then send this through to a 3D printer, and in a couple hours, we have a mold with our selected features printed on this device. We then take that mold and we mix it with a polymer known as PDMS. Now, PDMS is a biologically inert and optically inert polymer. It's actually the same polymer that contact lenses are made out of. So we mix this PDMS with this curing agent that forms a really viscous polymer solution, which we pour into these molds. We then cure and bake these at about 90 degrees Celsius to solidify the polymer. We peel out the PDMS polymer and we cut and trim this. We'll cut the device down. We'll punch in open inputs and outputs. We'll then do a wash to remove any uncured polymers, and then we'll bond this onto a glass microscope slide, followed by connection with tubings. And so what we're left with at this point is a PDMS micro device with a tubing input, a single channel along this glass slide, and an output. And so with this device, we then treat and seed this with an ECM matrix solution, which allows us to grow tissue culture cells on top of that glass microscope slide. So we could grow effectively any tissue culture cell line that we like. We allow the cells to seed under static conditions for 24 hours. We then start up a flow rate, and we eventually form a confluent epithelium, in this sense, a mucus-producing epithelium. And at this stage, we have an active epithelium that's producing and secreting a mucus layer, and we can inoculate this with a range of different microorganisms. This whole gut-on-a-chip device is kept inside a 37 degrees, 5% CO2 tissue culture incubator. This is the platform. We actually control the flow for this platform using a range of different pumps or syringe devices. This is a pump system where we have an air pressure source controlled by a computer flow regulator, which dispenses a set amount of air volume into a tissue culture reservoir inside the incubator, which will dispense media at a set flow rate, which will run through our device and into a waste container. These are some images showing you the main channel on the device. This main channel is about 1 centimeter in length and about 500 microns in width. These are some freshly seeded cells. You can see they're all spherical and very closely packed. We allow these to settle under static conditions for 24 hours. We form this sort of patchy epithelium. We then grow this under fluid flow for 2 to 4 days, and eventually we form this really, really beautiful mucosal epithelium that's just starting to differentiate. And you can see these sort of spherical structures that are starting to form these three-dimensional shapes, indicative of the types of crypts and villi and mucus secretion and production that you would see in the human gut. We're still a very long ways away from being a human gut, but it's a step towards that biological relevance. And so the real benefit, and this is our gut-on-a-chip platform. So this is a schematic for our device with our input, our main channel. It's about 1 centimeter in length, and our output. This is the device on a glass microscope slide and a picture of the cell layers and a 3D reconstruction of the device. So the idea is that we have our cells growing on this glass microscope slide, and that fluid flow across the surface of the device is going to create a constant shear stress and dynamic environment where the cells are continually producing a mucus layer that's eventually washed off from the device. And so using this platform, we can really use this as an experimental evolution device. And by inoculating in set concentrations of microorganisms, be that bacteria or bacteriophages, and that fluid flow, we can really force those microbes to attach, grow, and replicate within this mucosal surface. And effectively, any microbes that aren't growing in this mucosal surface are very quickly washed out and removed from this chip. So we effectively have a microbial incubator that's applying a really strong selection pressure to grow and replicate within this mucosal layer. Okay, so that's our gut-on-a-chip platform, summarized as briefly as I can. And so I want to go on to our experimental system. What did we actually do here? Well, we're using our gut-on-a-chip system, and we're inoculating and looking at the microbial dynamics between our T4 phage and our E. coli bacterial hosts. These are two model phage host systems that we use in the lab quite regularly. And T4 phage is a strictly lytic phage that will parasitize and infect E. coli bacterial host. So what we'll do is we'll take our fully functional gut-on-a-chip and we'll inoculate in our phage and bacterial hosts at a concentration of 10 to the 4 CFU or PFU per mil. And we'll run these under co-culture for 24 hours. So we're allowing the bacteria to grow and replicate, the phage to predate and replicate upon that bacteria, all within this mucosal environment for a 24-hour period. And one of the systems that we've built is a microbial sampling system that allows us to watch the growth and the ecology of the phage and bacteria within this system over a very high time resolution. So this is another example of our system. So our gut-on-a-chip controlled by a syringe pump controlling the fluid flow. And what we've done here is we've actually put a second chip on the end of this that sits on an 80-degree heat block. And so what's going to happen here is any of the microbes, the phages, the bacteria that wash out from this chip are going to pass into this second chip that's at 80 degrees Celsius. That temperature is going to inactivate the phages and the bacteria and stop them growing and replicating. We then dispense these using a dispensing robot into a 96-well plate at 30-minute time intervals. So every 30 minutes, that robot is going to move to a new well. And so we're able to collect 30-minute intervals of microbial DNA coming out of that chip. This is the actual system. It doesn't look as beautiful as the schematic, but here we have our syringe pump. It pumps via tubing into the incubator, which is pictured below here. So this is a side view of the incubator, and into our gut-on-a-chip system inside the incubator. We then have an exit porting coming back out of the incubator, back up on top onto an inactivation chip that sits on an 80-degree heat block to inactivate the microbes. And then this comes out into our dispensing robot, which is an XY stage connected to a Raspberry Pi controlled system that dispenses the liquid every 30 minutes into a new well. I've got to give a big shout out to Adrian Neal and Saab and Yachi. These are our engineering collaborators, and they've done a lot of help in designing this platform and our microfluidic system. So using this system, we can actually look at the high temporal resolution of both phage and bacteria coming out of our chip. And this is some of the data here. And I'm going to spend a little bit of time explaining this slide because it's really, really interesting. Another big shout out to Ciara Ket, who is a master's and now an RA student in our lab. And this was her project to build and design this system. So what you're looking at here is 3 individual gut-on-chip replicas, where we're looking at the phage and bacterial concentration in orange and blue, measured via qPCR. So this is log 10 copies of DNA on the y-axis, and we've got time over 24 hours in our 3 replicates. What we've done here is we've seeded the chips with phage and bacteria at that low concentration, and then allowed them to co-replicate and sampled every 30 minutes over 24 hours. And so what we can see in all of the chips is we see in the first 1 to 2 hours, a rapid crash of the bacterial population. Now, one thing I really want to highlight here is none of the samples have gone extinct here. These dotted lines are our limit of detection via our qPCR assays. So we see a crash in the bacterial population driven by the phages and a subsequent crash in the phage population. And then over the next 2 to 4 hours, we see the bacterial population and the phage populations recover. And then we see some really interesting dynamics. And I guess the take-home point here is that each chip is unique. We've done this multiple times and they all have unique dynamics. These are stochastic events that are not repeatable. In the first chip, we see the phages maintaining top-down control where they're really predating upon and controlling the bacterial population. We never really see the bacterial population achieve a high abundance in this replicate. In replicate 2, we see a complete opposite, where here the bacteria actually overtake the phage, potentially via phage resistance mechanisms, but importantly, both the phage and the bacteria coexist and persist in the system. And in replicate 3, we have these really beautiful predator-prey dynamics, where we have these increase in bacterial host, which is really quickly driven by an increase in phage, which subsequently drives down the bacterial host, and you have this classical predator-prey oscillating dynamics. So really interesting microbial dynamics. We haven't been able to investigate these at such a high resolution within these systems before. So there's a lot of interesting ecology that we can pull out from this platform. But really the take-home message is that each time we run one of these gut-on chips, it has its own unique microbial ecology, but that each chip can sustain phage and bacteria co-culture over at least 24 hours. Okay, so that's our platform demonstrating that it can support phage and bacterial co-culture. So what do we actually do in our experimental evolution? Again, we took the same similar system where we inoculated in T4 phage and E. coli bacterial host. We seeded the chips in triplicate at a low starting concentration. We ran these for 24 hours and we allowed the phage and bacteria to propagate and replicate upon each other. At the end of 24 hours, we collected all of the cells and all of the mucus produced within this chip, and we quantified both the phage and bacteria via CFU and PFU plating. What we then did is we isolated just the phage fraction, so just the phage population, which had previously grown and adapted for 24 hours in this mucus environment. We took this phage population and we seeded it into a new chip with ancestral bacterial host and ancestral mucus producing cells. And we repeated this experiment. We allowed this evolved phage population to replicate and predate upon the E. coli host within this mucosal environment. And then we did the same thing again. We selected this adapted phage population and put it back in with naive host and naive mucus cells. And so by this, we're evolving the phages over time within this mucosal environment. What we then did is we collected the phage populations from this environment. This is just showing you some of this data before I get onto that. So these are the counts that we got out from the chip. So this is our gut on a chip. Phage and bacteria concentrations on the y-axis, phage in blue and bacteria in red. This is after 24 hours. We can see again, as I showed previously, both of the phage and bacteria persisting. We've done this is replicate 1, where each of the phage population is transferred every generation out to 5 generations. And we can see that both the phage and bacteria persist in all of these generations, although there are some fluctuations in total abundances between the phage and bacterial host. We've also done this same experimental evolution setup, but in a test tube environment. So here there's no mucosal cells, there's no dynamic system or turnover. And here we see a very different microbial environment, where here the phages drive the bacteria to extinction, and we have a very high concentration of phages and low concentration of bacteria. We've done this over 3 replicates with 5 generations each. What we then have done is taken the phage population, sequenced the DNA via Illumina HiSeq, and then performed a BRESeq mutational frequency analysis to look at the mutations present within these evolving phage populations. So what did it look like? What was the data that we got back? Well, here is the de novo mutation frequencies that we saw arising within these gut and chip environments. So here we have 3 replicates, replicate 1, 2, and 3, 5 experimental evolution generations in each replicate. And the different colored lines are showing different types of mutations that are arising within this phage population. Now, I don't want to get into the mutations just yet. Rather, I want to look at the diversity and type of mutations that we saw between the gut-on-a-chip environment and the test tube control. And what I really want to highlight here is the total number of de novo or new mutations we saw arising. And the important thing is that we saw the same number of de novo mutations arising in the gut on the chip as we did in the test tube environment, really emphasizing that the mutational rate wasn't changing or increasing. Rather, we saw the types of mutations evolving in each different environment to be very different. In the test tube environment, we saw mutations appearing within the phage tail fiber and base plate domains, which is indicating of this arms race dynamics where the phages are adapting to better infect their bacterial hosts. Whereas in comparison to the gut-on-a-chip environment, we saw a lot more mutations in structural proteins, DNA and RNA binding proteins, and some hypothetical proteins, indicating these subtler mutations that were evolved within this mucus-like environment. Okay, so I want to look a bit closer at one of our gut-on-a-chip replicates. And this is the most interesting replicate and the most interesting mutational dynamics that we saw. So there were two other replicates, but for this talk, I'm not going to focus on those. So in this replicate, we've got 5 generations where we've transferred the phage population into a naive chip with naive bacteria and mucus, and each generation was replicated for 24 hours over 5 generations. The y-axis is our mutational frequency. We saw 2 major mutations arise within this chip, a mutation in the HOC outer capsid protein in blue, and a mutation in the GOF mRNA metabolism modulator. Now, I don't want to get into the function of these mutations just yet. Rather, I want to talk about their dynamics. And there's some really interesting mutational dynamics that we see emerging. First of all, neither of these mutations were present in our starting population. Yet after 1 generation, both of the mutations appeared and ranged between 25% and 40% mutational diversity. And this tells us a couple of things. This tells us, first of all, that both of these mutations likely emerged on their own. And what I mean by that is 2 populations of phages emerged, one carrying the HOC mutation, one carrying the GOF. And we can deduce this due to their mutational dynamics, due to the fact that both of them have separate dynamics and neither of them have crossed this 50% threshold. And in order to get over 50%, they would actually have to recombine or outcompete each other. What we see in later generations is exactly that happen. And around generation 3 or 4, we see both the mutation frequencies jump up above 50%. And importantly, both of the mutation frequencies co-aligned and actually persisted together. And what this is demonstrating is that these 2 mutations, which arose independently, have recombined and fixed onto the same genetic background. And if you think about this from a phage co-infection perspective, it's actually a really interesting question. How can 2 different phage genotypes recombine, lytic phage genotypes recombine? And the only way that this can happen is for these 2 phages to co-infect the same bacteria at the same time. We've illustrated this in our blue phage and our orange phage. Both phages co-infect a bacteria, and this allows a recombination event, and we get a recombinant phage genome that has both mutations, which replicates in lice and produces these progeny recombinant phages. Now, in order for this co-infection to occur, this requires a high phage to bacterium ratio. Now, if we go back to our phage to bacteria counts that we've seen on chip, we consistently see higher phage to bacterium ratios at the end of our replicates and in our temporal dynamics, suggesting that this gut-on-a-chip environment is really driving recombination as an evolutionary tool for these phages. So we wanted to test this in the lab. How do we go about doing this? Well, we took a mixture of 2 phage genotypes, our HOC mutant and our GOF mutant, which we mixed at a 1-to-1 ratio. We then took this 1-to-1 ratio mixture of phages and infected a bacterial host at a high multiplicity infection or phage to bacteria ratio of 10, meaning that there was 10 phages for every 1 bacterium. We did the same thing, the same mixture at a MOI of 0.1. We then allowed this phage mixture to go through a single round of replication, and then we plated those phage progeny onto a top agar assay, and we screened individual plaques for recombination. And what we found was that our high phage to bacterium ratio, or high MOI of 10, the recombination frequency was over 40% within a single generation, compared to our low phage to bacterium ratio, which had only 2% recombination ratio. Again, suggesting that these high phage to bacterium ratios are driving recombination, and that this is a major evolutionary mechanism that phages are using to evolve. Okay, so that's the first point. Now I want to dig into, what are these mutations? What do they do? Why are they interesting? Why should we care about them? The first one I want to talk about is the GOF mRNA metabolism. And what we found here was a 21 base pair deletion in this GOF gene, which encodes a transcription anti-terminator that prevents phage transcript degradation. Now, what is this mutation doing? We don't actually know. We tried a few different things. We've looked at growth curves and tried to play around with the function of this, but it's still a work in progress. We don't know the function of this GOF mutation. However, the HOC mutation we know a huge amount about. And actually, this was a lot of my postdoc work, was characterizing the role and function of this HOC gene in phages. And so I'm going to try to summarize a couple of years of my postdoc work in one slide. So this is the bacteriophage adherence to mucus, or BAM model. And so what we know is that epithelial cells and surfaces, particularly within the gut or the lungs in the human body, will produce a mucus layer. And indeed, the cells that we're using in our gut on a chip system produce an active mucosal surface. And what we showed in this work was that particular phage populations, and our T4 phage population was one of these, particular groups of phages were able to stick to and adhere to mucosal surfaces. And they were able to do this by a really novel and interesting binding interaction between immunoglobulin-like domains, or Ig-like domains, displayed on the phage's capsid or head, which allowed these phages, these domains, to form weak binding interactions with the glycans or sugars present within the mucus network. So the cells would produce this mucus, which was coated in different sugars, and the phages would stick to that mucus. And by sticking to that mucus, those mucus sugars, we formed a wall or a concentration of phages growing in that mucosal network. And when a bacteria tries to invade or colonize that mucosa, it's hit by this higher concentration or wall of phages, which very quickly infect, parasitize, and kill that bacterial host, thereby releasing more phages back into the surrounding mucosal environment. So we theorized in this paper that these mucus-adherent phages would have a selective advantage by sticking it to this mucosal layer, thereby allowing them to persist and replicate, and that there was a selective advantage to evolve and adapt to this adherence. And that was one of the ideas testing behind this project. Okay, so let's go back to our HOC gene. What did we see? What was the mutation, and how does this relate to the mucus? Well, we detected a single nucleotide polymorphism, or SNP, in the HOC gene in one of our chips. This mutation was a non-synonymous change from an aspartic acid to an asparagine amino acid at position 246, amino acid 246, which changed this residue from an acidic to a basic side chain. And we refer to this as the D246N mutation for this amino acid change at this position. And at its frequency, it got to about 80% of our population, highest frequency in our chip. So where is this D246N mutation present within the HOC gene? This is a schematic of the T4 phage capsid or head. You can see it's GP23 and 24, its major capsid proteins in blue and green, and the HOC protein shown in red. So that each T4 phage has 155 copies of this HOC protein that physically cover and protrude and stick out from its capsid. Each HOC protein is composed of 4 domains. The 4th domain, shown at the bottom here, is associated with bonding or sticking the HOC domain to the capsid. So this is the glue that sticks the HOC to the capsid. It's not structurally resolved, hence its globular shape. The other 3 domains, domains 3, 2, and 1, each encode for an immunoglobulin-like domain. And these immunoglobulin-like domains form a long, flexible filament, linear filament, that protrudes out from the phage capsid, with each of these Ig domains having been shown to facilitate adherence to mucosal glycans. So what we found was that D246N mutation fell within a beta sheet within the Ig-like domain residue 3, which was a predicted site for glycan adherence. So we had a mutation in the Ig domain associated with our phage, which was known to facilitate adherence to mucus. So we wanted to test, well, does this mutant actually cause a change in binding to mucosal glycans? So to do this, we used a technique known as surface plasmon resonance, or SPR, to assess protein glycan binding interactions. The first thing we did was we recombinantly expressed both our wild type and our HOC mutant phage along with a his tag. And this his tag allowed us to purify these proteins while also immobilizing them to a glass microscope microarray slide. So we had one slide coated with our wild type HOC protein and one slide coated with our mutant HOC protein. What we did is we washed a whole bunch of glycans across that, and using this SPR laser technique, we're actually able to measure the binding strength. How well do those glycans bind to those 2 different mutant proteins? And so this is the result from the SPR. So on the x-axis here, we're testing 26 different glycan types. And I'll just draw your attention to the different structures. We have a range of fucose structures, galactose, sialic acids, gangliosides. And on the y-axis here, we have the dissociation constant, or the KD. Now, this is measuring the detachment rate, or the strength of detachment of those glycans away from the protein. So it's a little bit counterintuitive, but a higher KD, or a higher dissociation constant, indicates weaker binding. What we're comparing here is our wild-type HOC protein in gray to our D246N mutant in blue. And so we can see a couple of things. The first thing that I want to highlight is that we only saw binding to fucosylated glycans. And that's a really interesting point that I'll touch on the next slide, but we saw no binding to any of the other glycan structures. We also saw that in some instances, both the mutant and the wild type had really strong binding to some of the glycans, but in other cases, so it's example 5, the mutant phage had actually a much weaker binding than the wild type phage. So this mutation was causing phenotypic changes in the strength of binding to the glycans. Now, I want to touch on these fucose glycans. These are really interesting. And so fucosylated glycans are really relevant to the human body. And so glycosylation is a process using variable enzymes that attaches a whole range of different glycans onto the structure and surfaces of your cells. And these are mediated by enzymes called glycosyl transferases. One really important glycosyl transferase in the human body is known as FUT2. And this encodes an alpha-1,2-fucosyl transferase. And it's highly active in mucosal tissues. Now, what FUT2 does is it attaches a terminal fucose sugar, shown in this yellow triangle here, to a growing glycan chain. So your body will produce these growing glycan chains, and FUT2 will stick a fucose on the end, and that will cap the glycan and end the structure. And so what's interesting is that about 20% of the human population lack a functional copy of this FUT2 enzyme. And as a result, they can't produce these fucosylated glycans. And these individuals are known as non-secretors. Conversely, secretors can produce fucosylated glycans. And there's a lot of interesting research coming out showing the big biological differences between secretors and non-secretors. And I'll just give you 3 examples of that I've taken from the literature. The first is associated with rotavirus infection. So rotavirus actually recognizes fucose, and that allows rotavirus to bind human intestinal epithelial cells and cause disease. So secretors that produce fucosylated glycans are more susceptible to rotavirus, whereas non-secretors are much more resistant to rotavirus infection. Another example is seen in breast milk. And so when secretors breastfeed, they will secrete fucosylated glycans in their breast milk. And when the infant ingests those fucosylated glycans act as a decoy and prevent bacterial pathogens from invading and attaching and causing disease. So rather than these bacterial pathogens binding and attaching to the infant's intestinal wall, they'll bind to these breast milk fucosylated glycans, which will act as a decoy, and that will prevent them from getting colonized and sick from some of these enteric pathogens. And finally, there's a lot of gut microbiome work in showing that secretors and non-secretors actually have very different gut microbiomes. So it's really interesting to theorize that these phages that can stick and adhere to mucus that are persistent within your gut are only sticking and recognizing these fucosylated glycans and may only be active in patients or people who are secretors. Okay, so I'm going to show you one last experiment before I wrap up. And so really, this is the final experiment. So to recap briefly, we've evolved a phage in a mucosal system. We've seen that this phage accumulated a mutation in its HOC gene, which is associated with binding to fucosylated glycans in that mucosal surface. And so we wanted to see, did this mutant phage actually outcompete the wild-type phage in our gut-on-a-chip environment experiment? So we designed a really simple experiment when we thought about it, but it turned out to be quite complex. So I'll try my best to explain it. So what we did is we took a 1-to-1 mixture of our wild-type HOC T4 phage, or we took our D246N mutant that we isolated from our gut-on-a-chip. We mixed these at a 1-to-1 ratio, and we washed it in the presence of 2 glycans. One of the glycans was a control glycan that didn't have a fucose, which neither of the phages bound to, so neither of them interacted to the control glycan. Or we washed it in with the fucosylated glycan that, if you remember, the wild type bound really strongly to, but the D246N mutant didn't interact or interacted much weaker. And so the idea here is that we'd wash these two phages at a 1-to-1 mix in the presence of this fucosylated glycan. Because the wild type phage would bind more strongly to the fucosylated glycan, it would be prevented from accumulating within the mucus layer in the gut on the chip. Conversely, a mutant D246N phage, which didn't bind as strongly to the fucosylated glycan, would accumulate and persist at a higher concentration in the gut on a chip device. We then did a washout for 4 hours, and we sampled the number of phages coming off this device. And the hypothesis was that if the mutant was less competitively inhibited by this fucosylated glycan, that we would see more of the mutant phage washing out than of the wild type at this 1-to-1 ratio. I hope I explained that well. It was a little bit confusing, but this is the data that we saw. So here you're looking at phage per mil washing out of our gut on the chips. There's no bacteria here, just phage over a 4-hour period. And we've got our wild type phage in gray and our D246N mutant in orange. And this is in the presence of the control glycan, which neither phage interacted with. And here you can see over 4 hours, both of these curves sit right on the top of each other. We have a high detachment rate of both phages, and towards the end, both of the curves are really fairly similar. Compare this to the fucosylated glycan, and we immediately see some differences. Again, the wild type in gray and the D246N in blue. First of all, we see a higher number of phages detaching from the chip in the presence of this fucosylated glycan, indicating that there was a competitive inhibition of our wild type. And then towards the end of the chip, towards the 4-hour, where we've got quite low phage counts, we see the wild type almost washing out to extinction, whereas the D246N was still persisting, indicating that yes, this D246N had a difference in binding to these glycans, and that led to an increased persistence within our gut nut chip. Okay, so let me bring this all together. So what were our conclusions from the study? Well, we're concluding that phages and bacteria can stably coexist in our gut nut chip device, and that this is a useful platform for studying gut phage microbial ecology. We also showed that the gut mucus layer drives the evolution of de novo mutations within these phage populations, and these were very different to our test tube control environments. We also showed that recombination is a major driving factor for phage evolution, particularly in environments where there's high phage to bacterium ratios. We demonstrated a mutation within the Hox structural protein that led to altered phage adherence to mucosal glycans. We ultimately showed that this provided the phage a fitness benefit that allowed them to persist within that mammalian mucus environment. And so we're pitching this as a novel trans-domain evolutionary axis where phages can co-evolve with their bacteria, but they can be selected and influenced by the gut mucosal layer. And so putting this in the context of the human body, we know that the human gut biome is incredibly unique to every person that we look at, and we believe that these evolution events are occurring within your phage populations, allowing the phages in your gut to adapt and persist directly to your own cells, organs, and systems of your body. This work is available on bioarchives. We are currently under review, but if you're interested about it, you can read some more online. I'll wrap up with some acknowledgements. Again, a big shout out to Wei Ho. A big shout out to my lab. This is a bit of an older photo. I'll give some thanks to some of our collaborators, Joe and Oren at Griffiths, who did all the glycomics work, Adrian Neild in their lab, who did all the engineering and gut on chip work, and Mike McDonald for his help with the evolution. Big thanks to our funders. And one last promo before I go. Recently announced we'll be hosting the Viruses and Microbes 2024 conference in Cairns, Australia. We're just in the preliminary stages of organizing this, but this is one of the premier phage conferences. It's going to be the first time the conference leaves Europe, and we'd love to extend a warm welcome to anyone here who's interested, and we hope you put this on your calendar and hopefully see you in 2024. With that, I'll wrap up. Thank you all for listening. Happy to take any questions.
Thank you so much, Dr. Jeremy Barr, and the discussion is open. Anyone who is interested in any question, please, they can go ahead.
Dr. Jeremy, this is Dr. Sanjay from Amrita, Kerala.
Hi, Sanjay.
Yeah, hi. I have been following your work very closely since your BAM model in PNS, and we did follow your work, particularly with our system, where we showed this phages bind to other matrix proteins, for example, fibronectin, collagen partially, and heparin. I was wondering your fucosylation, did you check with other proteins other than mucin?
That's a great
Fucosylation mutation.
Yeah, that's a great point. So I'll say two things to that. The first one is that you're completely right. These interactions are really promiscuous. The HOC protein, for instance, will bind a whole range of different glycans and other substrates. So there's a lot of different environments and substrates these phages could bind to. In terms of the glycosylation, this was some glycan work done by our collaborators at Griffith, and so we only really looked at these glycan structures. So some of the glycans were found in other environments other than mucin, in the human body or other environmental samples, but we only looked at glycans. We didn't test fibronectin or any other protein or macromolecule substrates.
Any on sialic acid binding?
Yeah,
So
really.
that's one of the sialic acid.
Great point. We didn't see anything, which was really surprising. That would have been my first guess. And when we got the SPR results, which is the first time we've done this, and it was really eye-opening, we only saw interaction with fructose residues, which was really, exciting and interesting. So nothing with sialic acids, which I was very surprised by as well.
Yeah. Thank you so much. It's a really, really great work. I really appreciate that. It's a breakthrough. Yeah. Thank you.
Hi, Jeremy. This is Urmi from Delhi.
See you again.
Yeah. So no point saying you do fantastic work, and I was really trying very hard to grasp everything which you were saying, because it's a lot of information condensed in a short time. Yeah. So fantastic presentation. I just had two quick questions. One is this gut chip platform that you've developed. So, is this prototype totally available to you, or do you think it'll be available for other researchers as well, if they do the same thing?
Great question. Yep.
And second is a simple one, like you said, that if you have higher phage to bacteria ratio, then there are higher chances of recombination. So usually, I think in the gut we have 10:1 ratio. Is that correct? That's the ratio of phages to bacteria. Yep. And you said 20% of the population do not have this fucose transferase enzyme. Do you also think that diet may be impacting the presence of fucose in certain guts, because maybe they have the enzyme, but fucose is not there? I'm just wondering.
Great question. So I'll answer your first one regarding our gut on a chip system. So there's a number of groups around the world that are developing this platform. Our goal is really to have it as a platform that we can study microbial ecology and look at how phages and bacteria interact in a gut environment, because it's very difficult to do this in an animal model, let alone a human system. So we're trying to develop this and move towards an anaerobic system. We hope to have this available. They are quite technically challenging, but definitely we're open to collaborating and working with people. Onto your second question, I think I forgot the first point, but the second point was regarding fucosylation and whether or not diet affects this. Diet will definitely play a large role. My understanding, though, is if an individual doesn't have an active copy of the FUT2, that they won't be able to secrete fucosylated glycans regardless of the diet, because it's actually the human enzymes that will metabolize and add on those glycan structures which are exported onto the cell surface.
Thank you. And one of the undergrads in my lab, he's working on a phage which he discovered, and he happened to find fibronectin domain in the tail fiber. So just wondering what its role is. So maybe he can get in touch with you to learn.
Definitely. Yeah, I'm happy to email and chat about that further. There's lots of really diverse ways that phages interact with these cells and surfaces. And the more we look, the more we find, which is really interesting and exciting to hear.
Thank you so much.
You're welcome.
Could I ask a question, please? Mark Tolman in Cardiff. Good morning, good afternoon, good evening, wherever you are. Jeremy, really fantastic talk. Thank you very much for that. I just wanted to ask a question regarding you're working with E. coli. Have you looked at invasion of your epithelial cell layers with E. coli and them hiding in there?
That is a great question. We haven't is the easy answer. The strain we're using is a very benign, E. coli B lab strain. That being said, one of the aspects that we really want to push with this system is working with more relevant gut microbes and pathogen systems. So we haven't looked at invasion yet. As far as I'm aware, these lab strains won't invade intracellularly, and we can co-culture them for about 36 to 48 hours. And the system fails then, but I think that is more due to the overgrowth of the bacteria and the tissue culture layers just simply not getting access to their nutrients rather than any pathogenic effects of the cells. We have tried to put some clinical Pseudomonas strains on the chip, and they just obliterate the cell layers within hours. So it's a really interesting question. We haven't done much work in that space yet.
Oh, okay, just following on from that. So have you looked at all at any other E. coli types apart from your lab strains?
Not in the gut on a chip. No, we haven't.
Okay. Yeah.
But it's something we're interested in expanding. And yeah, definitely.
Yeah, it's a really interesting system, and yeah, fantastic. Thank you so much.
Thank you. You're welcome.
Hi, Jeremy.
Hey, Ramesh. Good to see you.
Yeah, it's excellent work. It's impressive. And thank you so much for your time.
Thank you.
No questions.
No worries. Thanks, Ramesh.
Thank you.
Ask one question.
Very much for accepting our invitation. Thank you very much. Thank you, Jeremy. Since we are running out of time, we'll go for a next speaker.
Yes.
Delegates, if you have any questions, later, the email will be shared to you. You can be direct in touch with Jeremy. Thank you. Thank you. Thank you, Ranjita.
Thank you, Dr. Jeremy, for the useful insight. We now move to our next speaker, and it is my very great pleasure to introduce the president of... Hello. It is my very great pleasure to introduce the president of Society for Bacteriophage Research and Therapy, one of the pioneers who took phage therapy to the next level, one of the most knowledgeable and experienced phage scientists, my own mentor, Professor Dr. Sanjay Chibber. Dr. Chibber is presently the Emeritus Scientist at Department of Microbiology, Punjab University, Chandigarh. He was among the few selected for this high honor appointed by Indian Council of Medical Research as ICMR Emeritus Scientist, making him the only one from universities of India to have found a place in the list of 11 selected scientists for this award. Professor Chibber has more than 42 years of teaching experience. He has guided many PhD students, including international students, and completed many projects funded by ICMR, DBT, DST, CSIR. Sir has close to 260 international publications, numerous patents, book chapters, editorials to his credit, and the editor and reviewer of a long list of high impact journals. He has also served as the coordinator of UGC SAP program, Research Promotion Cell, Punjab University, DST Purse Grant, etc. His research interest has been into looking alternative strategies to curb drug-resistant infections, and his dedicated focus on exploring phage therapy and all its aspects speak their own story. In 2019, he was also awarded the very prestigious Lifetime Devotion Award for his contributions in the field of medical microbiology by Society of Microbiologists India. Sir, words fall short to summarize your achievements, honors, and your contributions to science and to the society at large. May I now request, sir, to enlighten us all with his words of expertise. The topic will be focusing on potential application of phages in treatment of secondary bacterial infections in patients with COVID-19 infections. Over to you, sir.
Thank you, Sandeep. Thank you very much for the nice introduction. Can you see the slides?
Yes, sir. Yes, sir. Yes.
Should I start?
Yes, sir, please.
Thank you for giving me the opportunity to speak on the potential application of phages in the treatment of secondary bacterial infections in patients with COVID-19 infection. In fact, the two talks before this have been for the understanding of the fundamentals of phage biology, but this is going to be different. And so I don't have much of my own work, but I'm talking about how the phages can find an application in treating the secondary bacterial infections. My talk is divided into two parts. The first part will be focusing on the secondary bacterial infections in the COVID patients. And second will be how we can use phages as antibacterial agents to take care of this particular infection. We all know that COVID-19 is the biggest pandemic of our time. In fact, it is the worst human tragedy which we have faced in the recent years. The virus has infected 185 million people worldwide, and it has caused 4.18 lakh deaths from COVID-19 in India, the official figure, between January 2020 and June 2021. The disease has impacted all walks of life, be it the healthcare, the education, the trade and economy, and politics. If we look at the burden of the disease in India, India has the second highest number of documented cases in the world at 3.1 crore. We are just behind US. But if it comes to the mortality, then India has the third highest number of documented deaths behind US and Brazil in the world at 4.18 lakh deaths. But the lessons which we have learned or we are going to face is that COVID-19 is going to stay here. So we will have to look for the means how we can live with this particular viral infection. As I've already said, it has impacted our lives in countless ways, but humans are resilient and they are gradually adapting to new normal. I'll just give you one example over here that work from home may not be the solution for everyone, as most workplaces will continue to have the bulk of employees carrying out operations in the usual way. But it holds our hope that those who could not aspire to work in the normal way due to the movement constraints will now have options. The job flexibility has assumed new directions, though no doubt there are shortcomings which need to be sorted out. Another point is that, however, the COVID-19 has accelerated the adoption of many new technological advances. These technological advances were available earlier also, but we were not using them as such. I'll just give you an example in education itself, because I'm from the education field. We never had virtual classes, virtual conferences, virtual meetings, but now this has become a part of our teaching. Therefore, you can see that COVID-19 has brought about changes in our day-to-day life. So with this introduction, I move to the secondary bacterial infections in the COVID-19 patients. It has been recognized for a considerable time period that the viral respiratory infections, they predispose the patients to the bacterial infections, and that these co-infections have a worse outcome than that of the either infection on its own. The best studied in this regard has been the influenza infection, where with the documentation in several epidemiological and microbiological studies, most of the deaths occurring during the 1918-19 influenza pandemic were due to secondary bacterial infections rather than the effect of inherently hypervirulent strain of virus causing a rapidly progressive fetal pneumonia. And in my next slide, I'm comparing, there are certain differences if we talk about the secondary bacterial infections in the influenza vis-a-vis the COVID-19 patients. And based on the bacterial cultures, the clinical parameters, and death rate, the following differences have been seen. These include COVID-19 patients. They have higher rates of bacterial infection than the influenza patients. Number two, time from bacterial admission to bacterial growth are longer in the COVID-19 patients. Number three, the late infection, of course, in both the influenza and the COVID-19 patients, the gram-negative bacteria are the ones which cause the secondary bacterial infections, but the late infection with gram-positive bacteria are more common in the COVID-19 patients. But one thing which is common is that in both the secondary bacterial infections, they are at the people are at a higher risk of death. Now, second component is that death is significantly associated with clinical parameters in the COVID-19 patients, but not in the influenza. And here I give you the example that the older people were found to be more susceptible to the COVID-19 infection. And this may be explained on the basis of the immunosenescence, which is a process of immune dysfunction that occurs with age. And it includes a remodeling of the lymphoid organs, leading to changes in the immune function of the elderly people and bringing them to the development of infections, autoimmune diseases, and the malignant tissues. Secondary bacterial infection is a notable complication which is associated with the worst outcomes in the COVID-19 patients, and this is based on the recent reports which have appeared. I'll be discussing a few of them. The nutshell, it is the careful surveillance and the prompt antibiotic treatment are beneficial only in the selected patients. And the reason may be the drug-resistant bacteria, because many such cases have been reported. Therefore, the alternate treatment strategies for the resistant strains need to be looked into, and we will talk about this in my second part of the talk. Now, looking at the secondary bacterial infections, which occur as a result of the upper respiratory viral infections. The common organisms which are responsible are the Streptococcus pneumoniae, Streptococcus aureus, Streptococcus pyogenes, because these bacteria are the one which colonize the upper respiratory tract. In influenza, up to 25% of patients, they develop the secondary bacterial infections, usually the pneumonia. As far as SARS is concerned, 25% of cases have secondary infections. This information is only based on one study, whereas in the COVID-19, the bacterial co-infections, they occur in 15% of all cases, including 50% of the non-survivors. Now, based on the experiences with other viral infections, the question is whether this novel coronavirus could be associated with the co-pathogen. And this question was considered important to answer because widespread antibiotic use in the hospitalized cases with COVID-19. And this is reported in the literature and the time when there were few publications regarding the co-infections and the superinfection. Furthermore, early guidelines for COVID management also recommended early use of antibiotics in all suspected COVID-19 cases immediately within 1 hour of identification of sepsis. Some of the literature renewed suggested that co-pathogens in COVID-19 cases were encountered in those who were severely ill and those who died. But these appear to be mainly because of the superinfections rather than the co-infection. And I would like to differentiate between the superinfection and the co-infection. And according to the CDC, the superinfection are infections following a previous infection, especially when caused by microorganisms that are resistant or have become resistant to antibiotics used earlier. On the contrary, the co-infection is one occurring concurrently with the initial infection. So here our emphasis is on the secondary infections rather than the co-infection. The findings of pathogen alone or together with a low lymphocyte count in addition to elevated levels of D-dimers were shown on stepwise multivariate regression analysis to be associated with severity of the COVID-19 infections. Now, the mechanisms which have been suggested as far as the development of secondary bacterial infections is concerned, many of the studies they have reported these mechanisms, which mainly include destruction of the respiratory epithelium, and it exposes the basement membrane, and there is an upregulation. Along with this, there is an upregulation of the molecules that bacteria use as receptors, especially due to the neuraminidase activity. And both of these, they increase bacterial adherence to the respiratory epithelium. The impaired function of the immune cells, including the neutrophils and the macrophages, the macrophages is affected by the release of interferon gamma produced during the T cell response to the influenza that impairs the clearance of the pneumococci from the lungs by the alveolar macrophages. But in case of the COVID-19 infections, the impaired ability of the host to clear the bacterial pathogens is related to the production of the cytokines such as the IL-10, IL-6, IL-17, IL-23, and along with this reduced dendritic cell function, macrophage, natural killer cells, CD4 positive and CD8 positive T cells, and also certain phagocyte independent mechanisms by which the virus infection may not be cleared immediately or early. So this is the mechanism which has been suggested. So in a nutshell, we can say that the downregulation and differential regulation of the immune genes are the mechanisms that may create a positive environment for the establishment of the secondary bacterial infections following the bacterial attachment to the host structural cells and the pro-inflammatory environment, which is conducive to the suppression of the antibacterial host defenses. Now, overall, if I have to summarize, this is the immunosuppression. This is the inappropriate use of antibiotics, which leads to emergence of multiple drug-resistant microbial pathogens in the hospital environment. And this, coupled with the prolonged hospital stay, this exposes the patient to infection with multiple drug-resistant secondary bacterial infections. And as I've already said, the immunosuppression is also likely to trigger activation of the quiescent biofilms in cached airway pathogens. This is then very difficult to clear this biofilm infections using the antibiotics. The fluid-filled alveolar tissue also is an ideal habitat for the bacterial growth for pathogens. So this was all about the antibiotics, but we also talk about the downside of the antibiotic biocide use, where it has been shown. I'll explain this to you in my next slide. Overuse of antibiotics and biocides can also lead to the selection of the resistant strains. Use of antimicrobials have also been advocated for prophylaxis or as repurposed drugs to treat COVID infection itself, even without a co-infection. And therefore, in this slide, I'm showing you that in recent years, or maybe during the COVID pandemic, we have used a lot of biocides, the antimicrobial soaps, the disinfectants, the antiseptics, the sanitizers. They can have two-way effect. One is induced we are using at a sub-inhibitory concentration, and this will lead to the under the selective pressure of these biocytes, there will be a selection of the antibiotic resistant organisms. And the second is that these biocytes will flow into the hospital waste environment treatment system. And from there, these will be released in the environment, maybe the soil and the manure, the surface in the underground water, the food or the plants and the crops, and terrestrial and aquatic animals. So basically with this slide, I'm trying to show is that one is the hospital environment, but this other is the environment as such, where also we might end up having the selection of the antibiotic resistant organisms under the selective pressure of these biocytes. So this is all about the COVID infection as such. And now I'll be concentrating on some of the reports which have shown that the co-infection upon admission is in the range of 3.1 to 7%, whereas the secondary bacterial infection following hospitalization, they range between 15 to 20%. And here I'm quoting one of the Indian study which has recently been published by the ICMR, in which they have shown 3.6% patients, they developed secondary bacterial or fungal infection. In fact, this is the data which has come from the study, which included 17,534 admitted patients to the 10 hospitals, the 10 hospitals of the Indian Council of Medical Research, and they looked at the incidence. The 3.6% they developed secondary bacterial infections or the fungal infection, and in around 20% in the admitted cases. The mortality, however, was very high, to the tune of 56.7% among these patients, as compared to the 10.6% overall mortality rate among the COVID-19 patients. But looking at the etiology of the infections, which is reported in this study, the Indian study, the gram-negative organisms were the predominant, constituting about 70% of the total isolates. And out of this, the Klebsiella pneumoniae was the predominant, 29%, along with the Acinetobacter baumannii, 21%. And together, both these pathogens, Klebsiella pneumoniae and Acinetobacter baumannii, constituted about 50% of the isolates obtained from the COVID patients. Around 35% of the patients showed polymicrobial infection, but the only important part which is there in the study is, one of the important parts which is there in the study is that these gram-negative organisms, they showed high carbapenem resistance to the tune of 92.6%. Now, possibly the Klebsiella, the gram-negative pathogens which were seen predominantly in these patients may be contributed to the use of steroids in these patients, or this may be because of the prolonged stay in the hospital exposing them to the nosocomial pathogens, or it could be the invasive procedure or the devices which were used in these patients, for example, the mechanical ventilators, or it could be the central or the venous catheter implants. So this possibly could have led to the infection by the antibiotic resistant strains in these gram-negative bacteria. There are many international reports which are available, but I'm just quoting one which has recently I have seen. This is the Dr. Vance Fowler, who said during a June 30 media briefing, which was sponsored by the Infectious Disease Society of America. He quoted that a man in his mid-40s, married with a small child, developed COVID-19 in the early January 2021. He was intubated and it spent about 1 month in the ICU and managed to survive. But since then, he has been stuck with a series of progressively more drug-resistant bacteria, This is the information from the Professor Fowler, who is Professor of Medicine at the Duke University School of Medicine in Durham, North Carolina, and chair of the IDSA Antimicrobial Resistance Committee. According to him, the patient acquired Pseudomonas ventilator-associated pneumonia while on the ventilator. Although the infection initially responded to standard antibiotics, but he has experienced relapses over the past few months. Through these multiple infections, the Pseudomonas grew increasingly pan-resistant to the treatment. So now we have a situation where we can say that antimicrobial resistance threat, it continues during the COVID pandemic, and therefore there are the stark realities of the antimicrobial resistance, which we have just discussed, including the rising rates of difficult to treat infections, a lack of robust pipeline of future antimicrobials, and COVID-19 treatments that leave people more vulnerable to infections. This is the urgent priority according to the experts. The second point is, for some patients, the pandemic and the antimicrobial resistance are intervened. Now, I'm coming to the second component of my talk, where I can say that the bacteriophages can be a potential agent to treat these secondary bacterial infections. And I'm saying so because with bacteriophages as therapeutic agents over the past few years, I've witnessed that these bacteriophages are very, very effective when we have the well-characterized lytic bacteriophages and use them when they have a broad host range. Their efficacy has been proven both in vitro and in the animal models of various diseases, be it the respiratory tract infection, the wound infection, the blood infection, the urinary tract infection, and so on. And its efficacy has also been shown in human subjects, and in fact, it has been sometimes used also to cure the infection. Ability to disintegrate the bacteria in the biofilm mode by depolymerase producing phages has also been emphasized, and the results in my study in our studies have shown that if depolymerase enzyme is given along with the antibiotic in vivo also, this makes the antibiotic more effective by disintegrating the biofilm. Yes, synergistic effect is also very important. The antiphages can be used in combination with the antibiotics, and it has been seen in that in many cases, it results in a synergistic action. And therefore, there is a need to identify the antibiotics which can basically act in a synergistic mode along with the phages. Of course, time of institution of phage therapy is very, very important. The earlier it is instituted, it is more effective vis-a-vis instituting at a later stage. Now, the question is, why use bacteriophages as therapeutic agents? The ability of the bacteriophage to rapidly kill or lyse infected bacteria, their specificity, and their proven clinical safety makes them ideal, robust, safe, and effective self-replicating antibacterial drugs. And I've given you in this slide, I'm showing you the various positive points: effective against multiple drug-resistant microorganisms. Number two, they are self-replicating, which means a single dose can result in increase in the concentration vis-a-vis the antibiotics, where the multiple doses have to be given. It is highly specific. It does not affect the normal microflora. The phages are easy to isolate. They are well tolerated with negligible side effects till today. They are quickly adaptable to combat the emergence of newly arising bacterial threats. They exhibit better pharmacokinetics than antibiotics, and they are also useful in patients who are allergic to antibiotics. Having said this, I would also say that there are certain therapeutic challenges with phages. Of course, the research is going on the world over, and a lot of issues have been almost tackled or will be tackled in the future. And these include the narrow host range, which may not be useful in cases of systemic infection. The phage immunogenicity is a source of concern, and experiments in our laboratory have shown that this can be taken care if phages are put into some nanocarriers or lipoidal vesicles. The genome of the majority of the phages has been unraveled, and now attempts are being made to do this. Lytic phages induce rapid lysis and release of endotoxin. This is a matter of subject, and the scientists are looking into how to take care of the endotoxin, massive endotoxin release on phage therapy. Resistance development is another issue. And the most important is the phage therapy as such cannot be patented, because as I said, these phages are being obtained from the environment, from the sewage, and such biological materials therefore are not patentable. So this is another issue which has to be taken care. But to overcome this, the scientists in the recent years have suggested the use of phage endolysins or the depolymerases. If we look at the phage endolysin, they have a broad host range, and they have a specificity depending upon the molecular characteristics. They kill the cells without entering the cells. So therefore, the bacteria are less likely to develop the resistance. Systemic biology can be used to increase their specificity. Pharmacokinetics well characterized. And since these are the biological entities which are being manipulated within the lab, and therefore such things or such endolysins can be patented, that is the positive point. So similarly, depolymerases, as I said, a lot of work is going on these enzymes, hydrolytic enzymes, and people are looking into how depolymerase can be used to disintegrate the biofilm, because majority of the chronic infections in the human beings have been related to the depolymerase production, especially in the biofilm mode. And therefore, once depolymerase is used, we have shown this in our experiments, this disintegrates the biofilm and exposes the bacteria which are embedded within the polysaccharide mesh of the biofilm to the antibiotics, which otherwise are not on their own can penetrate inside the biofilm. So these are the newer additions, the phage products, which are receiving a lot of attention, the endolysins and the depolymerases, and therefore they can very well be used against the secondary bacterial infection producing bacteria. There are a lot of interest is there. I've just given you a list of the major market players all over the world, the pharmaceutical companies who are working in this particular area, and soon we are likely to have many phage therapeutic products which can be of help to the mankind, and especially in the infections like COVID-19 infections, where the superadded infections, the secondary bacterial infections, are the cause of death in many patients. However, there is a need to encompass a variety of underlying opportunities that are required to be identified accurately in the global market environment. There exists a lot of factors for adequate market growth that are thoroughly inculcated in detail, such as the overall quantity of production and consumption, the gross margins, import-export, competitive landscape analysis, price analysis, diverse licensing strategies, graphical representation, essential vendor landscapes, market size, the total volume of sales and marketing, market regulation, because the regulatory authorities have to permit this, key investment areas, and key parameters for adequate market evaluation. So now with this, these words, I would like to say that the bacteriophage therapeutic market is going at a very rapid speed the world over. Many of the pharmaceutical companies, they are at developing the products which can be used against the drug-resistant bacteria. And we have a hope where we can think of that there is a future for the bacteriophages, maybe on their own or maybe in conjunction with the other antibiotic agents, the other antimicrobial agents. And therefore, we can say that these secondary bacterial infections in the COVID-19 patients, they develop a hope within us that these can be used. And however, the regulatory agencies of the country and the world need to look into all these areas. Thank you very much. And if any questions, I would be happy to answer.
Thank you so much, sir, for the very useful talk on a very important and relevant topic in today's situation. The forum is now open to any questions and any discussion for the discussion.
Hello.
Yes.
Hello. Thank you, sir, for the nice presentation.
Yeah.
Yeah, sir, I just want to ask that can we take account into the role of steroids into the secondary infections, as a variety of steroids such as glucosteroids, corticosteroids such as dexamethasone are aggressively used in treatment of COVID infection. So can we take them into account for considering the secondary infection pertaining to the COVID-19 infection?
Yeah, that is why this is true that the steroids, they lower the immunity of the patient, and which exposes them to the bacterial infections or the other infections. And that is the reason that the emphasis is that steroids only be used under the guidance of a clinician, and we should not just go blindly for the use of steroids as such. If these are needed, then only these be used. Otherwise, they expose us to many other problems as well.
Okay, sir. Thank you, sir.
Welcome.
Hi, sir. It was a wonderful presentation. Prashant here.
Yes.
So, what is your views on using bacteriophages in prophylactic treatment? Can it be used for prophylaxis?
We should avoid basically for prophylaxis, primarily because, as I said, this can, if we are not using in the proper dose, then this can lead to the development of drug resistance. And again, this should be used under the supervision of a clinician, even if we have to use it for a prophylactic purpose. Care should be taken not to overuse them for a longer period of time.
Okay, thank you.
Thank you so much, sir, for taking out your time and giving us this useful insight. I think we can move to the next speaker for the session. Thank you once again, Shibir sir.
Yeah.
I have a question that I can quickly ask. Sir, I mean, do you think that given the rise in secondary infection, secondary bacterial infection, ICMR is considering phages as potential therapeutics? Have you been part of any such discussion there?
No, I don't think so. The phage scientists have to bring this into the notice of the people who are sitting at the ICMR, because nobody have thought of, I think, for using phages. In fact, within the throughout the world also, if we can say, phages have not been considered up till now, and that is the reason that I'm saying that there is a potential, and we should think of using phages as antibacterial agents, especially in the drug-resistant bacteria.
Thank you so much, sir.
Thank you.
Okay, so moving next in today's session, it is my great pleasure and honor to introduce the next speaker, Dr. Vinod Kumar CS. Sir, you can stop the screen sharing, sir.
Me?
Yes.
Dr. Sanjay sir, yeah.
Okay.
Stop screen sharing. Yeah. Thank you, sir. Dr. Vinod Kumar is presently the Professor, Department of Microbiology, and also the Director, Molecular Lab, SS Institute of Medical Sciences and Research Center, Karnataka. Sir has published more than 130 research papers, and his research interest encompassed drug resistance among bacteria and yeast, phages as alternative to antibiotics against MDR infections, and phages as biodisinfectants and parasitic infections. With his expertise in AMR and phage therapy, it will be a delight to hear you, sir. May I now request sir to enlighten us? Over to you, sir.
Thank you, Dr. Sandeep. Can I share the slide? The slide is visible?
Yes, sir.
Okay, thank you. My topic of presentation is the new hope from a forgotten cure, hacking extensively drug resistance Acinetobacter baumannii infection using the bacteriophage isolated from an environment. My journey on phages since year 2000, it all began when I started doing my PhD. Could you please mute, Anu?
Participant, can you mute yourself?
May I request all the delegates to please put your mics on mute?
Admins, can you mute all except Vinod sir? Admin.
Can you hear me now?
Yes, sir. Yes.
Yeah, thank you. Thank you. My journey started in the year 2000 when I was doing a PhD on septicemia. It was a period we started isolating a lot of drug resistance, especially beta-lactamase mediated drug resistance among the escape pathogens. From then, we worked on the phages because it was a very difficult time. I was talking to my student a few hours back. Even getting a references on phages was very difficult because there was no literature as such available from Indian scientists, and getting a foreign references were very difficult. Since 2000, we have developed various models to work on the efficacy and the utility of the bacteriophages, mainly on the septicemic mice and the rabbit models on the drug resistance bacteria which cause septicemia in both pediatric and the adult age. Wound infection, that is a excision wound model, were developed in the mice and the rabbit. Burn wound model, and also the urinary tract infection, because E. coli, where the first or second speaker was highlighting how important the E. coli, and also I think the Mark Tolman will be speaking on the importance of E. coli and the drug resistance in UK. Apart from utility, the phage also could be very well used, and it is suited as a biodisinfectant. We coined this term biodisinfectant because we have a chemical, which acts as a disinfectant. So once you start using the chemical, you need to add, the extra dose of the disinfectant. But in this case, what we found in our research is, if you add a cocktail of phage and keep adding the specimen, the concentration of the phage keeps increasing and the bacteria are completely disinfected. Phages are also well suited in the sewage treatment plant because the chlorine which is used get easily oxidized. The free chlorine gets easily oxidized, and you need to maintain a constant concentration of the chlorine in the sewage treatment plant. So we used a cocktail of phages after studying what are the different types of bacteria which are present commonly in the effluent. And we added a cocktail of phages, and we could able to really appreciate the effectiveness of the phage in disinfecting the effluent in sewage treatment plant. The lambda phage is very well studied, and John, he explained about the two different, the choline and the anticholine aspect. And it has been very well studied in the lambda phage. The lambda phage could be used, and the chimeric phages could be easily developed because the lambda phage is extensively studied, and we know, like, all the genes what it annotates. So the fibers of the lambda phage could be replaced by the phage of your choice. We tried this especially for Acinetobacter and for the Pseudomonas, which can be easily used on patient. And we have characterized various phages for multi-drug and extensively drug-resistant bacteria, such as E. coli, MRSA, Acinetobacter, Enterococci, because the Enterococci and Staphylococcus, they are known to cause subcutaneous infections. And Proteus species, E. coli, Enterobacter, Klebsiella, they are known to cause UTI, septicemia, meningitis. And also the hacking of biofilm caused by these gram-negative bacteria using the cocktail of bacteriophages is very interesting. All those patients, who are ventilated or who are cathetered, they're known to form a biofilm on them. So it is always better to use, along with the nanoparticles, the phage can be impregnated on these devices, which can prevent the biofilm formation. These are the few major publications, since 2000. Klebsiella pneumonia meningitis model. This was mainly done on the mouse model where meningitis was induced and the bacteriophage were injected. The most interesting finding was the penicillin. The penicillin is a drug of choice in case of the meningitis, but the penicillin doesn't cross a blood-brain barrier. In such a case, we documented that bacteriophages were seen even in the brain of the mouse, indicating that they easily cause BBB. And Pseudomonas aeruginosa septicemic model in mouse and the rat. The most interesting finding was no neutralizing antibodies were documented because there were a lot of issues related with antibodies which could form in the septicemia, or the endotoxins which could be released during the septicemic infection. But naturally speaking, even under a gram-negative septicemia, when you treat a septicemic patient, the gram-negative bacteria, they are killed. And the endotoxins are released in the bloodstream. Body has its own mechanism to overcome these endotoxins which are induced during the septicemia. So in the septicemic model, we didn't document any antibodies production. Methicillin-resistant Staphylococcus aureus is a notorious pathogen in case of the diabetic foot, along with the Enterococcus species. So we could able to isolate, characterize the phages for MRSA and also for the Enterococci in the animal model, and it was as effective as the drug of choice. In this case, it was the clindamycin was the drug of choice in both the cases. It was as effective as the clindamycin. And Acinetobacter baumannii is a notorious pathogen which are intrinsically resistant to majority of the drug. In spite of having a robust hospital infection control policy, it is very difficult to contain the spread of Acinetobacter baumannii. So the phages are very effective against the Acinetobacter baumannii in case of uncontrolled diabetic model. And we even in silico analysis of a bacteriophage lytic enzyme was studied, and it is published in 2017. And as I mentioned earlier, the bacteriophage was far better than the chlorine in the sewage treatment plant. And there are a number of examples of the studies which suggest that chlorine is a carcinogenic. We should be very careful to use the chlorine in a higher concentration. So the bacteriophage could be an alternative to chlorine, and it can easily replace the chlorine in all existing sewage treatment plant. So you need not modify the sewage treatment plant. In the tertiary level, you can add the bacteriophages, and it is as effective or more effective and more safer, than the chlorine. Kinetic study was done because there are a lot of issues or the hurdles as a phage as a therapeutic agent, because there are not sufficient number of kinetic studies. Because yesterday talk, I've been listening, a lot of students were telling like how to fix the dose. So we studied this, which is published in 2020 and also in 2021. That's a modeling of carbapenem resistant Klebsiella pneumoniae. We tried with a different dose, assuming that, in any acute infection, there is a higher bacterial load. Keeping that in mind, we did a lot of animal studies, studying the lower concentration of the phage against the higher concentration of the bacteria, or at the same time, higher concentration of the phage with a lower concentration of the bacteria, or with the equal concentration. But what we found is the phages are very effective when the bacteria and the phages are of the equal concentration. It doesn't mean that, the lower concentration will not work. But the time taken wise, if you see, it is very effective when bacteriophage and the bacteria are in an equal concentration. Coming to the presentation, I have put the forgotten cure, the name as the forgotten cure. I would like to ask why phage failed, or why the phage is not accepted, or why phage is not still been accepted. In spite of having 130 years of experience on the phage and the data available in the public domain, the phage is not widely accepted because I personally feel, this is my personal opinion, we don't have a translational research which can take phage from the bench side to the bedside. Lot of research, lot of publications, lot of presentations, what we hear, what we read, they are more of academic interest only. Regulatory bodies are not accepting because we don't have those translational research data. I've just put 5 important points why or what are the hurdles, in preventing a phage as a therapeutic agent. The first hurdle is ecophysiological data on in vivo phage bacterial interaction in relevant animal model to select suitable phage for the clinical application is very, very minimal. We need to have a fundamental knowledge on the phage bacterium interaction in a pertinent animal model before successful clinical application be envisaged for the phage therapy. We don't have a proper animal model study. The second hurdle is polymicrobial infection should be avoided or addressed with a poly-pronged approach. Majority of the, I'm talking in terms of human infection, the wound infection, if you take it, is more of polymicrobial infection. So when you have a polymicrobial infection, there's no point in selecting a single phage against the polymicrobial infection. So the species specificity characteristic of most phages is generally higher desirable in monomicrobial diseases. However, this specificity can be a major limitation in case of the polymicrobial infection. So it should be geographically customized. Because assume, I have a diabetic foot infection with a polymicrobial approach, I can't just use those phages which are supplied by someone else. I need to identify and I need to have a cocktail of phages thinking. It is something similar to your antibiotic policies which are used at the institutional level. Even a phage policy should be customized to a particular geographical area. Sterility and the purity of the phage preparation. Yesterday, I think one of the core committee member, was asking about preparation and the purification about the endotoxin. So the therapeutic formulation need to be free from pyrogenic exotoxin and the endotoxin, because the preparation of phage itself will have a lot of endotoxin. Until and unless we have a robust system to take care of these endotoxin, or the preparation which is free from endotoxin or host cell protein or DNA or residual agent, it is not possible to use the phage preparation. Because what happens is rapid lysis of the gram-negative bacteria caused by phage therapy, accompanied by the endotoxin release, potentially causes pro-inflammatory signaling cascade system, which can lead to endotoxin storm, or it can lead to enhanced production of interleukin-6, leading to multi-organ failure. Hurdle 4, difficulties including the high specificity rendered by the bacteriophage targeting only few strains. So until and unless we select the bacteriophage which has got a genus specificity, it becomes very difficult. I'll give you an example. Again, take a diabetic foot, again, or take a urinary tract infection. So you have a different strains of E. coli in case of UTI, or different strains of MRSA. So you can't have a high species specific phages which can lysis only that particular strain of MRSA. You have to go for a cocktail of phages which can kill different strains of MRSA. Genetic modification of the host rendering its virulence and the pathogenicity. This is most important concern raised by the regulatory authorities. Now they keep questioning that until and unless the phage complete genome sequencing of the phages are not studied, it is not possible to allow the use of phages on the humans or as a therapeutic agents. They have no issues with using on inanimate object. You can use a phages in the STP, sewage treatment plant. You can use it as a biodisinfectant. But when you want to use it as a therapeutic agent, each phage should be extensively studied to show that none of the gene of the phages can code for any toxin. Because if you take an example of diphtheria, there's a Corynebacterium diphtheria which causes diphtheria because the toxins are mainly produced by the phage which has integrated with the host. So these are the main hurdles which we need to overcome, if you want to see the phages in the limelight. Coming to my presentation, this is mainly on the Acinetobacter baumannii. In 2000, if you see 2000 in fact, even till 2005, Acinetobacter baumannii or the Acinetobacter species were considered as a contaminant. Even if you isolate from any specimen, it was regarded as a contaminant. It was not considered as a pathogen because the Acinetobacter baumannii are normally seen in the soil. If you take a pinch of soil and you culture, you will grow the Acinetobacter baumannii. But the first publication was from Manipal, that is in 1996. My teacher, he published it suggesting that Acinetobacter is a pathogen, causing neonatal septicemia. So when I was doing my PhD in the year 2000, even I could able to document a lot of Acinetobacter species from blood collected from the neonates. And a lot of babies, they died because it was earlier considered as a contaminant. But these Acinetobacter species are intrinsically resistant to majority of the available drug. And as of today, they are all, I think the Mark Tolman, is going to speak on Acinetobacter baumannii, which is resistant to colistin. But in our center, we have not documented any of the Acinetobacter baumannii which are resistant to colistin. So we published, it was the second publication from our lab in the year 2004, suggesting that Acinetobacter is a potential pathogen causing wide infection. It could be a ventilator-associated pneumonia, as Dr. Sanjay Chibba was telling. In case of COVID patient, Acinetobacter baumannii is an important pathogen, aggravating the ventilator-associated pneumonia. So Acinetobacter baumannii, it can cause any disease. You name the disease, it can cause pneumonia, it can cause skin infection, soft tissue infection, wound infection, urinary tract infection, meningitis, bloodstream infection. you name it, Acinetobacter can cause infection in them. So you need to have a very stringent hospital infection control policies to overcome the Acinetobacter baumannii infection. In this regard, we isolated the phages because the hospitals have reported, coming back to this introduction, okay, hospitals have reported emergence of multi-drug resistance or extensively drug-resistant Acinetobacter species, and it also has become endemic. It is not from our center. Globally, people have started reporting the emergence of resistant to the Acinetobacter baumannii. Right? In India, if you see, in India, within 4 years, that is from 2002 to 2006, the antibiotic resistance bacteria, which were resistant to 7 group or the classes of antibiotic, they were resistant to more than 21 drugs. So in a span of 4 years, there was almost 3-fold increase in the drug resistance pattern. Even in Antarctica, people have started documenting or reporting the presence of antimicrobial drug resistance bacteria, even in the animals and water samples. And if you see at antibiotic or the new molecules of antibiotic, since 1990 to 2010, or of late from 2010 to 2020, hardly there are no new antibiotics because none of the pharmaceutical companies are coming forward, for production or discovering the new molecules, because huge money is spent on discovering. Before, the use of these antibiotics on the patient, the bacteria are resistant to these antibiotics because, as you are aware, the majority of the antibiotics are synthesized by the fungus. When you look into the ecosystem, the bacteria have evolved how to overcome these metabolites of the fungus. So what are the alternatives? Definitely, it is the bacteriophages. There are a number of other alternatives are available. If you just go to wound journal, you will find so many other alternatives for wound infection. People have come out with the natural remedies like neem oil. People have come out with the new remedies like using citric acid along with other ointment, for the utility. But I personally feel, in spite of other alternatives, as effective, the bacteriophage are more effective when compared to the other alternatives available. The most important point, this is a widely common, slides which you can see, but for those youngsters, I want to say these bacteriophage are highly species specific. How the key is specific to that particular log. There are specific phages. The phage which can kill E. coli may not kill Klebsiella. It may not take care of Staphylococcus aureus. So when you try to isolate, you have to see to it that the phages isolated are, the genus specific and the species specific. So when you look into the phage history, in 1896, Hankin, he said there are some invisible microbes which can kill bacteria. That's the reason, people who go and take the holy dip in River Ganga, they get cured with a lot of disease because it is said that Ganga water or the river Ganges has a lot of bacteriophage. So the society, society to honor the work done by these great scientists, they're going to give the awards for different scientists. Frederick Trott in 1950, Felix D. Harrell, he said, the phages are effective against the Shigella. George, Olivia, and Burrow, and a lot of scientists now, since 2000, we have thousands of Indian scientists who are also working on the phage therapy or the utility of the phage. What's the aim of my study or the presentation is the therapeutic potential of bacteriophage specific for extensively drug resistance bacteria. That's Acinetobacter baumannii, and ability to resolve the wound infection. Because why the rabbit model was taken? Because it replicates the clinical condition. And the efficacy was also studied in terms of epithelialization, wound contracture, general condition between the varied control and the other challenge groups. And also we assess whether there are any antibodies produced in the wound infection. So around 75 Acinetobacter baumannii were isolated, and a gene sequencing study was done, and the drug resistance pattern was studied, and it revealed that almost all Acinetobacter were resistant to almost all available drugs except the colistin. And when we characterized, it showed that Acinetobacter baumannii were ESBL producer, AMC producer, carbapenem resistant, and none of them were resistant to colistin. Student keeps questioning me, like, how to isolate the phage. Phage are present everywhere, but only thing, these phages are not specific to the bacteria. So you need to look for your specific phage from a different area, right? For my studies, I've been isolating phage from Western Ghats, from the river and the sewage system, and even, the Acinetobacter baumannii phages were isolated from the paddy field. Paddy field, we were isolated, and they were absolutely species specific. It showed 100% lysis characterization when it was challenged. The temperature, the temperature and the pH aspect was studied, because one of the presenter yesterday, they were telling that the phage they isolated was stable at the temperature 2. So, but it is very difficult to use such phages in the human condition, because we need to have a phages which are highly stable between 35 to 38 degrees centigrade. And we isolated the phage and we subjected the phage for Acinetobacter for the different pH, and we didn't see the phage recovery between the pH 3 and the pH 5. In the pH 6, we could see the concentration dropped when compared to the initial concentration, but the pH 7 and the pH 8 was very optimum, and the temperature was between 36 to 38 degree. The kinetic study was done in vitro by using the Spectromax molecular device, where we challenged the bacteria with the drug that's a colistin. We challenged the bacteria with the bacteriophage alone. We challenged bacteria with the bacteriophage and antibiotics, and also with a cocktail of bacteriophage apart from the control. And we could see that when the bacteriophage was used alone when compared to the cocktail, because here the bacteria used was a single bacterial strain, that's Acinetobacter, not a cocktail of bacteria. But we wanted to see what will be the effect, in vitro effect, when we use a cocktail of bacteriophage against a single. But the end result was almost same, when you use a single bacteriophage or a cocktail bacteriophage, when you want to challenge a monoetiology. But when you want to challenge a polymicrobial infection, definitely a polymicrobial cocktail of phage could be used. Now, this was a very interesting finding we could able to document. And we tried even in vivo also in the rabbit model, where the rabbit was, the diabetes was induced by injecting with the aloxon hydrate, and we divided 6 groups of 5 animal each. And the lateral left hind limb was shaved, and the area of 25 millimeter square was defined mark, and it was surgically cut under the ketamine anesthesia. And these are the different groups, as is said. So I'll just right away go to how the scoring was done. The scoring was done in the scale of 0 to 5, where 0 indicates the death of the animal, whereas the 5 indicates a normal and unremarkable condition, and between 2 to 4, it was a moribund state, or severe illness, a moderate illness, or slightly illness in case of score 5. And yeah, these are the animals. I'll straight away take you to the picture. If you see the normal control, this is the excision which was done on the lateral hind limb, where the area of 225 millimeter square was cut under anesthesia. And in case of the infection wound, a 10 to the power of 8 colony forming bacteria were sprayed on the wound. And the minimum lethal dose was evaluated with a different dose, 10 to the power of 10 to 10 to the power of 1 to 10 to the power of 10. And we found 10 to the power of 8 could easily induce a pyogenic infection within 48 hours. So we took 10 to the power of 8 colony forming unit as an infective dose. At the end of the day 2, that is after 48 hours, all those groups which were infected with the bacteria were challenged. This is the second group. I just take the color highlighter. Yeah, this is the second group. This is a group where bacteriophage, it's a challenge with the bacteriophage. And this is a group which was challenged with the antibiotic. And this is the group which was not challenged. It's a control group. And this is the group which was challenged with the antibiotic and along with the phage. So if you see the untreated group, at the end of the day 2, a lot of exudates can be observed. The wound did not heal up even at the end of 4, at the end of 6, and all the animal in the group that died at the end of the day 8. And the post-mortem of the animal suggests that the animal died mainly because of a multi-organ failure. Whereas when you compare that with the group which was challenged with the phage, at the end of the day 2, we could feel, you could see a lot of exudate here. Okay, this is the exudate, the pus oozing. At the end of the day 4 also, there was slight reduction in the pus formation, but at the end of the day 6, no bacteria were isolated and the pus was cultured from the wound, or the phages, phage titer started dropping at this point because no bacteriophages, no bacteria were isolated from this group. When you compare with the bacteriophage along with the antibiotic, the results were remarkable, but the bacteria were isolated even at the end of the day 8, suggesting that it took slightly a higher time when compared. Maybe the antibiotic played or hindered the proliferation of the bacteriophage. So when the bacteria were treated with the antibiotic along with the Cocktilia, this is a group which is showing the results were almost similar. And all these animals, they recovered completely at the end of the day 12, and complete epithelialization was seen at the day 28. If you see the wound contraction rate, the group which didn't receive the antibiotic, the contraction rate was in the negative trend. This is the red color, what you're seeing. This is the animal which was infected and unchallenged. All these animals, they died at the end of the day 4, day 6. Whereas in case of infected and challenged, even though the wound contraction rate, because at the end of the day 2, the wound contraction was in the negative trend because the phages were treated only at the end of the day 2. And later, the wound contraction was in the positive trend. This is what you can see. And all the animal recovered epithelialization was very effective. And we also evaluated the number of bacterial load and phage load at the different interval. So in case of phage treated group, the phage were isolated, in a higher amount when the bacteria were infected. When the no bacteria were isolated in that particular group, the phage concentration started decreasing at the end of the day 8, and the bacteriophage concentration was less than 100, and the no bacteriophage were isolated at the end of the day 12, indicating that when there are no bacteria present in the wound, so the phages will not remain, they get killed because there is no host for them to replicate. So this is a very remarkable or a very important finding, because people keep questioning, whether the phage will remain for a longer duration. The answer is no. When the host is not present, because it is the intracellular virus, it requires the host for the replication. If the host is not present, the phages, will be eliminated or cleared from the system. We also assessed the antibody in this wound model. We could not demonstrate any neutralizing antibodies, suggesting that, for acute infection, because this was observed till the 6th week. We collected blood at the end of every week, and we studied, we assessed for the neutralizing antibody by the plaque reduction assay, but we could not demonstrate any neutralizing antibody. So to conclude, there are a lot of findings, even in the literature, which suggest that phage could be a substitute for treating multi-drug resistance bacteria, or when there are no alternatives available. So I personally feel it is prudent to proceed with the phage therapy as a convincing data, and the kinetic data, safety data are available in the public domain. What is most important is, dialogue with the regulatory bodies and end user, which can facilitate the building of the future with the bacteriophage as an antibacterial agent when the impact of antibiotics are fading. And we have a second option, or the second window for a forgotten therapy. It would seem timely to begin to look fresh at this approach. Thank you very much. Such conference, gives a platform to join hands, to build a future of bacteriophage. Thank you. Thank you, Anand.
Thank you so much, sir, for the very excellent and highly informative talk, giving very useful insights into the prospects of phage therapy in treating drug-resistant strains of Acinetobacter baumannii, and with special emphasis on the fact that we need more animal models to support our findings. The forum is open for discussion.
Good morning. Thank you very much for your nice talk, Dr. Kumar. I would like to know what is the situation in India about the phage therapy in terms of laws. For instance, in Europe, it's very difficult to implant phage therapy in hospitals, but I would like to know who is the situation in India right now.
Welcome to the conference, Dr. Pailar. It's happy that you joined the conference. Can you come again, please? And I didn't catch you what you said.
I would like to know if it is possible to apply the phage therapy in your hospitals in India.
Because we are bound by the law of the country, the DCGI, there's a Drug Control General of India, doesn't permit. But if two registered medical practitioner, gives a permission that there are no other alternative available to treat the patient, yes, we can use phages as a therapeutic agent, but it is with the consent of the treating physician along with the patient attendant.
Okay. As you know, in Europe it's quite difficult to apply. For instance, in Spain, it's not possible right now. And I would like to improve this situation because we need phage therapy in hospitals, because we also have a lot of infections that are resistant to antibiotics. But it's quite difficult. We are fighting with the government to be able to implant the phage therapy here, but to the moment we are looking for that, and we hope in the future we can use phage therapy also here. Thank you. Thank you for your answer.
I totally agree with you, ma'am. Our Secretary, Dr. Gopal Nath, actually is in constant touch with the Indian Council of Medical Research and the Director General of Drugs. I think we would have got the permission, if the COVID situation wasn't there, because he has done a tremendous work on various aspects. Maybe shortly, when the COVID situation subsides, I think the phage could be used on patient. But phage for the environmental usage, we don't have any issues, ma'am. Thank you.
Dr. Vinod, this is Dr. Rao. I have a not a question, but an opinion on this.
Yes, Dr. Rao.
This is regarding the customized approach. So that is the most relevant approach in the future for the treatment. We need to go for like an antibiogram or like a phagogram type of approach. But for this, we need to have a repository of phages. Do you feel that there is a need for a centralized repository in the country so that anybody can access the phages? Like how we get the online antibiotics for this?
Very interesting question, Dr. Rao. The society, society as a repository, which is headed by Dr. Taruna, I think they have a repository, and I think there are few labs, I think headed by Dr. Ramesh, Dr. Gopalnath, and maybe even Sanjay, and even in my laboratory, we have a very good repository for the phages. Right, and we are in plan to have a centralized repository where the scientists can easily procure these phages. And as you rightly said, the antibiotic policy, similarly, the phage policy is also pertinent, when we start using the phage as a therapeutic agent.
Thank you for the clarification.
Dr. Vinod, did you find the bacteria which are phages? Have you studied if they become resensitive to antibiotics or the MIC for the antibiotics coming down on phage treatment?
Urmi, you're not audible. Dr. Urmi, can you come again?
Urmi, you're not audible.
What I heard is, have you documented phages resistant to any of the bacteria resistant to the phage? Yes, definitely. But the resistant in terms, we need to evaluate in terms of block diameter. What we have documented is when the first time when we did the block assay, the diameter of the block was very wide. When you lyophilize them and you reassess them after some time, the block diameter reduces. So when you keep propagating it, definitely the diameter keeps decreasing, which is similar to something like antibiotic sensitivity testing. Yes, we have documented. There are a good number of literature which suggests that bacteria can evolve over the phages.
Dr. Vinod, congratulations for such a nice presentation, and you actually showed the total your journey from beginning to till date, and it was really very impressive. And I hope that will be coming with something, and somehow we are getting the permission from the government of India at least for some use, or maybe like antibiotic policy, the fast policy is also being made. So this is very good idea. And let us keep working. As I talked to the DG ICMR, he told that you keep on working. So we are working, maybe in coming 4 or 5 years, we'll be succeeding in this area. So let us do it. And really, this is the very good, this conference going on. It is very successful and encouraging. And we learned a lot from the speakers and distinguished speakers from outside the country. And your presentation was really very nice. Dr. Shipper presented so well. So again, I thank you all, and congratulations for your nice presentation.
Thank you, sir. Sorry for the interruption. Vinod, sir, there are a few questions in the chat box. Can you see that and answer it?
Okay.
So I'll just tell you. There's a question from Rutuja. She wants to ask, when the mice were treated with phage and antibiotic together, were they added in together or in any order was followed?
Both were sprayed together, because for the wound infection, what we did, we sprayed the phage and we injected the antibiotic. It was done simultaneously. And one more question is if the clock diameter, no, it's a clock, PLAQ, clock diameter is wide, then bacteria is more sensitive. Definitely, yes. That is a normal principle which is used, clock diameter wide. And if you get around, 17, because I have seen around 30 to 35 millimeter, the phages which were isolated from the field against the Acinetobacter baumannii. Whereas the same phages which were isolated from the hospital effluent, they had a lesser diameter, indicating that the phages which were isolated from the hospital environment, the bacteria were resistant to it.
So there's another question. Is there any broad spectrum endolysin which is available that can kill both gram-negative and gram-positive bacteria? From Sashi.
A lot of scientists are working on endolysin. Definitely, people have come out with, especially Gangajan has come out with the enzymes which can be very effective against the MRSA. But as I said, it's the evolution bacteria try to overcome that. When you ask me whether the enzymes are better when compared to the live bacteria, the live bacteriophage, my answer is always the live bacteria, because it is a synergistic effect of the enzymes which are more effective when compared to the single enzyme. Or else you have to go for a cocktail of enzymes. You cannot go with a single enzyme. You have to go with a cocktail of enzyme to treat any situation. Now, I personally feel, going for a chimeric phage using the lambda bacteriophage will definitely take phage into greater height. Thank you. Thank you very much. If there are no other questions, we'll break for a lunch and we'll assemble back at 1:55 for one more very interesting topic. Thank you. Thank you very much. Thank you.
Thank you, sir. Thank you.
Thank you. Thank you, Sandeep. Thank you.